Mixed cannulae, expanding performance standards. High-performance cannulae for direct and femoral cannulation in minimally invasive cardiac surgery. LivaNova's complete range of mixed dedicated products allow easy and reproducible procedures. The portfolio includes the sutureless aortic valve Perceval, Memo 4D for mitral valve repair, the HeartLink system for assisting the perfusionist in applying goal-directed perfusion therapy, and a broad choice of high-performance dedicated cannulae. LivaNova's range of cannulae suit every cannulation technique for minimally invasive cardiac surgery. Venous and arterial femoral cannulation can be performed using the WRAP venous cannula and the EasyFlow Duo. For arterial direct and venous femoral cannulation, the EasyFlow aortic cannula and the WRAP are available. For venous femoral access, the LivaNova WRAP cannula features a dual-stage configuration for optimal drainage of the inferior and superior vena cava without interfering with the right atrium. The high-flow dynamics of WRAP improves the capability of drainage with a low pressure drop. An optional Sellinger dilator kit is available to perform percutaneous insertion. The LivaNova EasyFlow Duo cannula is designed for a safe and smooth femoral arterial cannulation. Its conical dispersion tip design allows a more gentle flow in the femoral artery, and its red cap avoids blood backflow during obturator removal. A dilator kit for percutaneous insertion is also included with the cannula. EasyFlow cannulae are designed for safe, reproducible, direct aortic cannulation and include a malleable stylet to aid cannulation insertion, facilitating bloodless introduction into the aortic arch, as well as a special conical dispersion tip for a more gentle flow in the vessel to reduce shear stress.
The point-of-care program at Huntsville Hospital is approaching its 20-year anniversary. As we began to grow as a hospital and provide more complex services to our community, the need for point-of-care testing became very clear in our organization. Many of our departments consider the EPOC device to be crucial to their ability to quickly move their patients from entry to the hospital to procedural room. It helps them monitor the patient's status during their procedures and post-procedure, so it's very integrated into our entire continuum of patient care. If you have any patient population where blood management is crucial, then a point-of-care device is very useful in those types of settings. It can provide a quick result on a very small amount of blood, and that is really important in some of our patient population.
We are starting a new process where we're going to screen our patients on the floor for sepsis. The patient can look and actually feel okay. They will show us signs and symptoms of sepsis. So what we do when they show these signs, we get a lactate level.
Lactic acid is a product of the sepsis cascade. So with sepsis, the inflammatory light switch goes on, the cascade starts churning, and unfortunately, once it starts going, we have to stop it. It will not stop on its own. The faster the lab draw, the faster the lab results, the faster you can act on it.
The process in the lab is what the process is. It just takes a certain amount of time for the instruments to run those tests.
I mean, we're talking 30 minutes even on a stat because that's the process. That's just how long it takes. There's not really anything you can do about that. For every hour you wait to give an antibiotic, the mortality for that patient increases 8%. So now that we have the handheld device, the nurse can get those results within a few minutes, and we know how to treat that patient. If a patient is going into septic shock, we do not have minutes to spare. We have got to get it done quickly. So when you get the little beep from the EPOC that says your results are in, you go with it. You don't even think twice about it. You're like, this is good. This is what I'm going to do for this patient. The next steps are clear.
I think our mortality on the initial floors dropped by almost 50% in the first 3 to 6 months. At this point, we're about hospital-wide. I think we'll be hospital-wide another 2 to 4 months. So this has become a huge program within our system.
Without the EPOC device, it would really be hard for us to get everything done in the timely manner. So it's really helped us to get our program going.
I mean, whether you're a surgeon, whether you're a medicine doctor, whether you're a dermatologist, you want to be able to do things in a timely fashion so that you can be most effective.
This really makes a difference in patient outcomes.
Like all perfusion professionals today, we are busy and frequently short-staffed. Does taking time off for a conference create a burden for your teammates? Has reimbursement for your continuing education dropped? Have you priced the cost of going to a meeting lately? Registration fees, airfare, transportation and parking, hotel, and meals out? This totals up to having an out-of-pocket expense of over $2,500. And then you hope the content is good. Perfusion Education has heard your concerns. They have a simple and seamless system that works on any platform, computer, tablet, or even your phone. You can chat during the program or even phone in and be live on the air. The content, second to none. Perfusion Education is by perfusionists and for perfusionists. Create a free account and check us out today.
Hello everyone and welcome to PerfWeb 34 Day 2. We have a very exciting and I think timely and provocative discussion program for you today, especially in light of everything that's going on with the COVID-19. But you'll see all of the parallels, I think, as we move forward. I want to go through the opening remarks very quickly if I can. I don't want to spend a lot of time on this. You got to thank our sponsors, please. Without them, we have absolutely nothing. We got to keep these lights on, and especially now with the squeeze of what's going on financially in this country, we need them more than ever. So really need you to consider thanking them, clicking on their sites and looking at what products they have that may help you in your practices. As you know, we're on all social media platforms. It's very important that you like us, follow us, share us, and give us the thumbs up, subscribe to our YouTube channel, and ask for notifications with the notification bell, please. Check out our websites at perfweb.us and perfusioneducation.com. Those sites have tremendous resources for you to earn all of your required ABCP CEU from one source so that you don't have to go anywhere. You know, it's more convenient, it's more affordable, and it's high quality education for you guys, covering a whole host of different topics with real content experts. We have a call-in number today. I really need you guys to write this down, 832-239-5358. When you see that sign, the phone lines will be open, and you can ask questions live on the air. Of course, you can always chat those questions via YouTube or via Messenger and Facebook, and also on Twitter. We're monitoring all three of those platforms right now, and I think LinkedIn as well. So any questions that you have, please go ahead and we'd love for you to call in, be a part of the conversation. Okay, I'm going to go straight into introducing what I consider to be probably one of our most incredible faculties that we've had all year. We've had a lot of good ones too, but to my right here in the studio with me is Dr. David Platt. Dr. Platt has a PhD in carbohydrate chemistry from Weizmann Institute of Science from Tel Aviv in Israel. It's in Tel Aviv, right?
Yes.
In Tel Aviv, yeah.
Yes.
And completed his postdoctoral work at Wayne State University in Michigan. He is a pioneer in designing drugs derived from copolymers. He has over 30 years of experience in drug development with over 30 patents, authored 10 journal articles, completed 2 textbooks, and I'm going to show you those books right here, which you can get from Amazon. I highly recommend it for those scientists out there. Has been the CEO of 5 public companies, 3 of which were on NASDAQ, and conducted over 30 clinical trials. He was the very first to combine chemotherapy with copolymer to target cancer, which radically improved the safety profile of the treatments. He coined the name galectin, which we're going to talk a lot about, and was the first to publish in a field of research that now produces over 300 peer-reviewed articles annually.
3,000.
His 3,000, I'm sorry, this says
Annually is 300.
300 annually, right, but a total of 3,000. Thank you, we appreciate that. His life's work is embodied in his latest invention called BXT25, which is an oxygen transport delivery drug that is 5,000 times smaller than a red blood cell and is capable of treating stroke, ARDS, and other diseases that have an element of hypoxia associated with the disease. Also coming to us, if we can bring Dr. Majewski up. Hey, Dr. Majewski. Dr. Majewski is
Hello.
a professor and research and does research and activates and I'm sorry. Dr. Majewski is a PhD and professor emeritus at Bar-Ilan University in Israel, doing research and teaching activities since 1965. Now, you don't look old enough to be teaching since 1965, Dr. Majewski. And,
Thanks.
and just if you could you move to your left 2 inches?
Pardon?
Move to your left 2 inches. Oh, that was
Like
about,
this?
yeah, a little less. Halfway. To the right now. Nope, the other way. To the right. Right there. Perfect.
Okay.
Now you're dead center in the screen. Thank you so much.
Okay.
His main field of research is brain and other organ physiology related to oxygen supply and metabolism in vivo. Professor Majewski has published more than 240 articles, as well as a book published in 2015 by Springer. Professor Majewski is the world leader in monitoring in vivo of physiological activities in the brain and other organs of experimental animals, as well as in human patients. He has developed many experimental models in animals related to changes in oxygen supply from hypoxia and from ischemia and its effect on mitochondrial function. He has served as a consultant to various pharmaceutical companies in evaluating efficacy and safety of new developed drugs. Professor Majewski was the chief scientist of 2 medical device companies that developed and attained FDA clearance for those particular products. Energy metabolism and function of the brain and other tissues in vivo under various pathophysiological conditions is his main field of research since he started his postdoctorate training with Professor Brighton Chance at the University of Pennsylvania in 1972. During almost 50 years of activity, he was able to accomplish a large number of goals that have been very significant, impacting on the understanding of physiological processes in the brain and other organs evaluated under in vivo conditions. The vision was to develop experimental approaches and tools that will enable us to study various physiological processes in experimental animals and at the same time will be ready for clinical applications. His philosophy has been to contribute significantly to society's health and well-being after enabling and funding his research activities during the last 5 decades. In addition to the large number of studies in experimental animals, he was able to translate the methodology into a platform of medical devices that will affect in the near future patients' outcomes in various clinical conditions. Just some of the highlights for you, Dr. Majewski, if I could bring it up, please. You've been the developer of the first fiber optic-based fluorometer reflectometer for NADH monitoring in anesthetized and unanesthetized animals. You developed a multi-parametric monitoring system for evaluating mitochondrial function and microcirculatory blood flow, something we've actually talked about. So I've got some questions about the microcirculatory blood flow and side stream dark field imaging. I'm somewhat curious on your opinion on that. and development of a new tissue vitality index, in quotes, vitality index, based on a large amount of experimental results and adopting of various modeling approaches. So you've done a lot in your 50 years, and Dr. Platt, you have done a lot in your 30 plus years. And I do, if I can you zoom in on these two books? I'll put them up right here. And I highly recommend you guys, I'm sorry, Dr. Majewski, I don't have your book, but these are two of Dr. Platt's books.
I will show it to you in a second.
Please, here, he's going to get it for you. But take a look at this. You can get these on Amazon. I highly recommend you get them and read them. They're very interesting, very provocative, very science-based. And there is Dr. Majewski's book. I highly recommend that as well. I need to get a copy for myself. So these are very interesting topics that do a lot of interesting things, especially in the field. So we're going to start off with the MDX viewer. Then we're going to talk about the nanomolecule and oxygen transport, oxygen delivery. And we're going to do the program today in three parts. Dr. Majewski first, followed by some discussion. Dr. Platt second, followed by more discussion. And then a combined discussion about everything we discussed. Then we're going to split the program. We're going to take a short break, and we're going to come back, and we're going to discuss some investment opportunities for both the MDX viewer and also the nanoparticle, so that everybody out there who's watching the program that may have an interest in this can do it. But we are going to separate it out from the science portion of it and the evidence-based medicine part of it, and then separate it to that, and we'll bring Mike Seek in as well to talk about some of those opportunities. So with that said, Dr. Majewski, we have your slides. They are up. If when you're ready to go to the next slide, just say next slide, and I will switch it for you.
Okay. Next slide, you can start, please. Okay, next slide. This is just the introductory one.
Mm-hmm.
I don't see your screen so clearly. Okay.
Here, we'll fix that for you, Dr. Majewski.
Okay, that's better. Mm-hmm. Okay, thank you.
Here we go. MDX viewer, the third vital sign.
Right. If you want to get to see the device itself, actually, you can see it behind me. Just look at behind me. There is the device is staying on the stage over there, so you can see it.
Yes, I see it.
Okay.
We see it. Can you make his screen bigger just to show him?
The only thing I can do, I think, to take the camera closer to the device.
There, we did it right there. Now we can see it. It's full screen.
Oh, okay.
And you're coming to us from Israel, right?
Yeah, I am now in Israel.
So it's about 10 o'clock at night there?
Yeah, now it's 10 o'clock.
We thank you very much for staying up so late to be with us today. Okay, so go ahead. So we'll go to the next slide, and we're ready to go whenever you are, sir.
Yeah. Okay, just to show an example, what is the need in diagnostic of patient is ARDS, which is one of the main clinical terrible situation in the intensive care unit and other department of clinical daily life. And this is ARDS, which is really a problem of the respiratory system. And there is not enough oxygen to supply to this patient because the lung is not working well. And actually today we face it when we are talking about the corona. So the main issue is to try to solve the problem and to evaluate the efficacy of oxygen delivery to the patient under this severe situation. Next slide, please. Okay, today, what the current measuring devices in the operation room and ICU and other departments in clinical daily life is either to use the pulse oximeter to measure the oxygenation of the blood, or measure the blood pressure that you see in the center, or to listen to the heart rate, measure temperature, and in the intensive care unit, of course, you have more parameters that could be monitored, but all of them are providing information about the systemic system, namely the cardiovascular system and the respiratory system. None of the parameters that are measured today are telling the physician any information about the tissue level physiology and biochemistry, which is the most important information that need for daily treatment of the patient. Next slide, please. As an example, you can see here, this is a brain of a patient that if you have a probe, you can put the probe on the brain, and the device will show you 4 different parameters that I will go in more details in a minute. You see it on the right-hand side of the figure. I just will mention it. We measure the metabolic state of the mitochondria regarding the oxygenation of the mitochondria and the cell, and we measure the blood oxygenation in the microcirculation. And the third parameter, the blue one, is the tissue blood flow, and the fourth one, the yellow, is the tissue blood volume. All these 4 parameters can be monitored by the MDX UL simultaneously from the same area of the tissue. So you can correlate between the 4 parameters and understand very well the physiological and pathological or pathophysiological events developed in the patient along the time in real time, second by second. Next slide, please. Now, the device that you see on the right-hand side, the MDX, that I showed you before in my room here, is measuring 4 parameters that you see in part E. The technology is an optical approach. We are using optical probe, using different light waves, different color of light. We excite the tissue, and then we can collect the light scattered back from the tissue and analyze it and obtain the 4 different parameters in real time continuously. So you can see in part B, you can see the main parts of the device that I will not go to details, it's more technical. Part C show you one of the main applications that we can use the device to monitor patient in the operation room and in the ICU. And here we want to use a very well-known pathological or pathophysiological event that develop in a patient when oxygen is not, the balance of oxygen in the body is negative, namely the supply of oxygen, like in hypoxia or ischemia, the amount of oxygen delivered to the brain or to the organs in general, to the body, is not enough. And then the body uses the adrenaline to shift blood from the less vital organ to the more vital organ, namely the brain and the heart are protected under these conditions, getting more blood, and the less vital organs like the skin, it can be the intestinal wall, and so on, they get much less blood. So if you put the probe, our probe, that can be embedded, as you can see in part C and D, it's a Foley catheter, urine Foley catheter that you can see it in outside the body, and here in C, you can see it inside the body. We measure the 4 parameters simultaneously from the wall of the urethra. Now, once you measure this parameter from the urethra, you are going to see an early warning signal coming from the body of the patient, telling you that there is a problem in oxygen supply to the body as a system. Therefore, the body shifts more blood from the less vital organ to the more vital organ, the brain. In order to preserve the organism, you need the heart and the brain to continue to work, even you can create damage to the less vital organs. So this is what you can see here, the basic concept. So when you put the Foley catheter in the patient, you can measure and you get 4 parameters that you see in part E that I mentioned before. And this tell you about the tissue level physiological parameters that telling you the oxygen balance in the microcirculation and the cellular compartment, namely the cells. In part F, you can see in the future we will embed in F and G, we'll have 2 more parameters that will be added to the 4 parameter that we measure with the Foley catheter. We will embed 2 more probes into the Foley catheter, so we'll get some systemic measurement via the Foley catheter from the same location in the urethra. Next slide, please. So actually, we are want to know what is the level of oxygen that is available in different compartments of the body. So if we start in air, the atmospheric air, we have about 160 millimeter of oxygen, partial pressure. In the respiratory system is about the same. In the cardiovascular system, it goes down to 95. You can see it on the second row. And then you have in the tissue microcirculation is going to 20, 30. And intracellularly, it's about 1 and 2 millimeter of mercury of oxygen. So the level in the cell is very, very low. Therefore, if there is a shortage of oxygen in the other system, like the cardiovascular and the respiratory system, the cellular area will be affected tremendously immediately when there is a shortage of oxygen to the patient. Therefore, we are looking on an area in the body that is very sensitive to the oxygen availability any second in the life of the patient. Now, on the right-hand side, you can see here what I already mentioned, that you have the systemic vital signs that's used today, and we are going to add the lower part of the scheme that you see the microcirculatory oxygen supply, and you have the tissue oxygen balance, which can be measured by the mitochondrial NADH redoxate, which is the enzyme that helps to create the ATP in the mitochondria in order to supply the ATP as a source for energy for the activity of the body. Next slide, please. Okay. Now you can see the details of the 4 different parameters. I will go very shortly, not too many details. But on the left-hand side, you can see the mitochondrial function measured by the fluorescence of one of the enzymes, the key enzymes in the respiratory chain of the mitochondria. And on the right-hand side, the upper part is the tissue blood flow. In the lower part, you see on the left side, the blood oxygenation and the tissue reflectance on the right-hand side lower. Now, the blood oxygenation that you will see here is not the same as the pulse oximeter is measuring in the patient today. We are looking here on the oxygenation at the level of the microcirculation in the capillaries area and the very small arterioles, which is much lower than what is available in the large arteries. Therefore, the measurement of this parameter is important to add to the battery of parameters that we measure together. So actually, by using these 4 parameters, all of them are optical techniques, we can get a very good picture of what happened in the microcirculation and in the cellular compartment at the same time. So we can now move to the next slide. Here you can see the balance, what we are calling a tissue oxygen balance between oxygen supply and oxygen demand. Normally, there is a very small reservoir of oxygen in the tissues because the tissues are dependent all the time on continuous supply of oxygen via the circulatory system and the respiratory system. Therefore, the reserves in the tissue is very low. Once you stop the supply, you get a shortage of oxygen in a few seconds, and then you start to go into a problem of oxygen supply. So we want to know all the time the balance between the supply and the demand. So if the demand is going up by activation, like the muscle, if the muscle is working harder, you want to give more oxygen, you will accelerate and give more supply. But if the supply is limited, then if you increase the demand without changing the supply, the balance will become negative. And if it's negative, then the tissue go to hypoxia and lack of oxygen and all the damages that can create to the tissue by shortage of oxygen. Therefore, it's very important to keep the balance between oxygen supply and oxygen demand that actually enable to produce enough ATP that you can see here in the center by the mitochondria. And most of the ATP that produced in the cell is dependent on the oxygen availability and the mitochondrial function. Next slide, please. Okay, here you can see the Foley catheters that I showed you already before, and the technology a little bit in more details, but written here. You can see here the power supply of the system and the bedside unit that is in behind me. You saw it before. What we have different light sources that it's embedded in a light source unit, and all these wavelengths that we have to excite the tissue with are coming from this unit. Then the light is going via the Foley catheter down to the urethra wall, and the light is coming back in fiber optic channel, another channel in the fiber optic probe that you see here, and it's collected to the detection unit, and the detection unit differentiate between the 4 parameters and calibrate it in relative units, and we can see it display on the device in real time. Next slide, please. Now, here you can see as the next generation that we actually already start to work on it, and we publish on it a few papers already. And this is to translate the real-time data that we collect from the patient using the MDX device, and to calculate a more easier parameter for the physician to interpret the physiological situation of the patient. Namely, we take the 4 parameters that are monitored by the MDX viewer, and we integrate it into what we call the tissue metabolic score, the TMS. The tissue metabolic score is actually a mathematical calculation based on thousands of animals that we used in the past, and also about using mathematical models. We found a way to calculate the 4 parameters to a single sort of a score that the physician can use. Once the score is going up, it means that the oxygen supply, oxygen balance is okay. If it's going down, it means that there is a shortage of oxygen and the balance is negative, and the patient will go into a pathological state development. So that you can see on the left side of the slide. On the right-hand side of the slide, you can see the systemic patient monitoring system that already exists, and then you can calculate from these parameters that monitor from the patient, you can calculate also a sort of an index or a score, and then later on to combine the 2 scores together into a machine learning device or system expert system, and get sort of a signal that will tell the physician when the patient become ischemic or hypoxic, namely, once there is a shortage of oxygen to the body and the system, it means that it will take few more seconds or minutes to get damage to the brain and to the heart. So by looking on the urethral wall via the Foley catheter, we can get the information earlier than it will appear in the brain, and we can, the physician can make an action to protect the patient by a different kind of procedure, for example, giving more oxygen or injecting a drug to increase blood pressure or whatever is needed. So this kind of early warning signal, this is a dream that actually will get through the clinical daily work if the device will be sitting next to each patient that need real-time monitoring of the oxygenation of the body of this patient, like in the ARDS or other kind of situation, severe situation, that oxygen supply to the body and the system is disturbed by different kind of pathological states. Okay. And actually, this slide is too many details to go step by step, but this shows you, in general, that the MDX has a very high value as compared to what's today available in the market, namely medical devices that can compete with the MDX. Actually, if you look on the right-hand side, where the MDX is compared to all the parameters that show in the second and third and fourth row, all of them have some green check and some X, red X. But if you look on the ICU device or the tissue metabolic score, you can see that all of the parameters that we are measuring, we provide information about the microcirculation together with the intracellular compartment, the mitochondria. So all is checked with green. So this is the main advantage of the MDX over all other kind of devices available in the market. All of them are based on one or two parameters. We are the only device, we have the only device that can measure 4 different parameters together simultaneously in real time, coming from the same tissue volume. Therefore, the value of this kind of parameter is tremendous for clinical daily diagnostic and follow-up of efficacy of either drug giving, like to giving the BXT25 to provide more oxygen, or to give any other drug to the patient. I think we are almost, this is just a short of potential application. You can see that mitochondrial function is so basic in the daily life of each tissue, each cell of the body. Therefore, if you can monitor the mitochondrial function in real time, you can actually provide information that will save many lives and improve the prognostic of each of the patient that comes through different clinical situations. I think that's the last slide, right? Okay. This one.
That's the last slide.
That's the last slide?
Yeah,
Okay,
okay.
very good.
That's the last slide.
That show what mitochondria are measuring oxygen,
Yes,
different disease indication.
yes, yes. That was this slide here, right. So very good. Okay, so so we want to go to full screen now? Yep, very good. Thank you so much, Dr. Majewski. That was intriguing and very interesting, and I would say it makes perfectly good sense to me, and I believe
The device, by the way, was approved by the FDA.
It's going to make, so it is approved by the FDA.
By the FDA.
So, because it makes a lot of sense. I wanted to ask you, I've got a series of questions that I want to ask you about this. Simplistically, if I can, and Dr. Platt, you feel free to add into this as well, you essentially get either a score or an index that is positive or negative. You either have sufficient supply or maybe even luxurious supply, or you don't and it's negative. Is that sort of distilling it down to what the tissue oxygenation score is?
Yeah, it's not the supply, but it's the balance between the supply and the demand.
Balance. Yes, supply versus demand, essentially.
Right.
So what you're saying is that this is going to be a real-time, second-by-second window into the homeostasis of the patient in regards to their oxygen supply-demand ratio, and much faster than waiting for a lab where you're doing a surrogate, a lactate to infer that you have some type of ischemia going on somewhere.
Yeah, lactate, for example, that you mentioned, the lactate delay in responding is it can take minutes, maybe 10, 15, 20 minutes before you start to see a real change in lactate. Now, lactate is a parameter that's not related only to the oxygen supply, but it's also related to the balance in the body of all the hematological situation of the patient. So it's not so specific. We are looking here with the MDX, we are looking on very specific parameters that related to the oxygen supply and demand directly. Therefore, we have compared the lactate, and always the lactate is delaying in response by 10, 15, 20 minutes after we can see the change with our device.
Very good. So, I have to ask, just to be sure, specific. So the device does not necessarily selectively measure. It's very global. So if there's an issue, you have to identify where that ischemia is coming from. Is that correct? Unless it's it's global, general.
Yeah, it really depends. We didn't go into it in the presentation, but it really depends where you put the probe. For example, if you want to measure a patient in a neurosurgical operation procedure, during the operation, you can put the probe, and we have it on one slide, we show the probe on the brain.
Yes.
If you put the probe on the brain directly, you will measure exactly what happened in the brain and not in the body.
Gotcha.
But if you put it in the urethra using the Foley catheter, the picture will represent the body as a system in addition to the urethra, because the urethra is a less vital organ, is a very early responder before the brain and the heart that are protected in the initial state of the pathological development. Therefore, it depends where you put the device.
That makes perfectly good sense to me. I completely understand that. Now, you clearly know a lot, and I think are recognized in the field of hypoxemia. Can you maybe just give us a little bit of more awareness of what you've been doing with your life to become recognized in this field?
Well, actually, when I started to measure the mitochondrial function, that was one parameter that I measured at the time. And we look on the mitochondrial function in real time, and that's I did during my postdoc period. But then I found that the physiological systems are so complicated that if you measure one parameter or two parameters from the subject, from the tissue in the animal or in the human, the information will be very, very limited in possibility to interpret the real pathological event. Namely, that if you use one parameter, this is the worst case that you can analyze any pathological state in the patient or in the animal. The more parameters that you add, the better the diagnostic tool you have in hand. namely that we start with 1, and then we add 2 and 3 and 4. We came in the brain to measure 8 parameters in a patient in the intensive care unit. So we have a probe that was sitting on the brain of a patient in the ICU, the neurosurgical ICU, and we follow up this patient for 48 hours continuously, and we were able to indicate exactly what happened every minute, every 20 seconds, whatever time you want to select, but very short. And we found, for example, in this case of a patient in the ICU, the neurosurgical, that this patient that we were monitored, the brain became after 24 hours, it became damaged. The brain became damaged because oxygen supply was not enough, and we saw the changes. But the definition of brain death was not clear yet to the physician. My device was able to show the events in the brain that indicate that this brain is now in a brain death situation. And it happened about 1 hour in this specific patient, 1 hour before the clinician were able to define and determine brain death of the patient. Now, why is it so important? First of all, you can take more measures to try to help the patient if you do it earlier. Secondly, if you want to have an organ donation from such a patient to use it in transplantation procedure, the earlier you know when the brain is dead and you can start the procedure for donation of the organ, you can get a better organ for the donation.
Oh, that's very
true.
So
there's many advantages to do this.
That's very true, because the heart actually suffers damage after brain death while we're waiting to explant it for the transplantation. That's very true. So I see a lot of opportunity in terms of the use of this type of technology. Have you explored the use of this device, the MDX device, in the urethra during the MDX viewer during cardiopulmonary bypass? Have you used it for patients who are transitioning from, let's say, pulsatile perfusion to mechanical, their native circulation, to mechanical circulatory support through either the heart-lung machine or ECMO or something like that, where you now have continuous flow, no pulsatility, and a lot of hemodilution and a lot of microcirculatory derangements that occur? What's your experience with that that you could describe?
Yeah, in this kind of procedure of the cardiovascular operation, we have only 11 patients that we were able to do statistic analysis, because you know that each patient has different timing, and to get statistics you need large number of patients. But even with a small group of 11 patients, when we analyzed the data, we found something very clearly. I don't have it here on the presentation, but what happened is that once you start the operation and you open the chest of the patient, some patients start to respond immediately when you start the procedure, when you open the chest and you start with the surgery. So other patients may start the response in the urethra once you start to put him on the heart-lung machine. Now, what happened is normally in the group of the patients that we did, all of them show that during the operation, the blood flow to the urethra went down to almost zero. The mitochondria was dysfunctioning completely. It stopped working, the mitochondria, in the urethra. And it stays the same level of low flow and high or low mitochondrial function until the patient's chest was closed and sutures and come back to a normal activity after, of course, disconnecting him from the heart-lung machine and close of the chest. So we have now a very good idea that during a severe stress that the patient have, you will go into a situation that the urethra can give you the needed information. And now, why you need it further. Now, if you take the same patient from the operation room to the short ICU intensive care unit for the cardiovascular surgeries, you can follow up to 24 hours post-operative period and see whether the patient is stable, whether the patient is deteriorated suddenly, or if the patient come out from the operation not in a good situation, is he improving? Can you do something to help him to improve? Now, all is based on what you will see on the urethra parameter or the index.
That makes good sense to me. Dr. Platt, do you want to add to this? Because I have a couple of more questions, but I haven't heard from you yet, and I apologize. I get to asking questions and I can't stop.
That's very important for the reason we're here, because the Professor Majewski actually demonstrate the future, how you can measure and predict organ performance. That doesn't exist today.
Yes. So getting to that, how, Dr. Majewski, Dr. Platt, is this device commercially available? It's FDA approved, or the technology is, but is it available?
No.
And
we need,
if it's not, why not?
We need about, we calculate about 2 million to make it in production. So we are on the stage that we're looking for to collaborate with the company.
Hopefully some companies are watching, but we'll talk about that then after the broadcast. I didn't understand the reason that the only thing that's keeping this from actually being in the market
it's money.
Is money. Okay, so we can talk about that a little later. If you're a investor or you're a company representative that may be watching, please watch the end of the program. It's going to be important for you. So obviously heart surgery, any major surgery, trauma would be appropriate. ICU sepsis, I would think. You know, I wanted to talk to you about side stream dark field imaging, because I have done that. I have looked sublingually with the viewer, and when we go on the bypass machine, I can see with my own eyes the microcirculation and the capillary density essentially disappear. And so being, of course, but it's I mean, it's important to see it. I think there are two different technologies, right? It would have been super if I could just stick something under the tongue, or in this case, something in the urethra, because essentially that ridge is just, the capillary bed is very close to the surface. You can see it. Here it's the same thing.
This technology is the future in many ways. Imagine that in the form that it's now, we need to do beyond commercializing that and put it in ICU or any ambulance or any emergency or any situation that you want to know in advance that organ going to fail.
That you're having an oxygen deficit of some sort. You're having a metabolic derangement is occurring secondary to tissue hypoxemia.
Exactly. So the future is, as you take that technology in this case, it's in a large, electronic form, and you miniaturize that, make it very small, and you can put it on a smart watch. And now you measure the state of hypoxia constantly, not heart beat, not blood pressure. We're talking about the state of hypoxia for every person. So there's a constant monitoring of homeostasis and tissue health of the entire body.
Well, when you go and you look at, Dr. Majewski, you're probably aware of this. I know you are too. You go back and look at all of the previous work done by Schumacher on optimizing tissue perfusion and survival in sepsis based on oxygen delivery. And so, I can't remember the term that was used for that. What's the term that was used for that? I can't think of it now. But nevertheless, you want to hyper oxygenate the tissue. You need to give the patient volume. You need to get the patient's cardiac output up because it's all about DO2 and getting super high levels of oxygen to the tissue. But a lot of times with our normal parameters, and you have a patient that just looks kind of wimpy, you really don't know what's going on, but you give a little of this and you give a little of that, maybe you put a balloon pump in, and well, your blood pressure is okay, it looks okay. They're not failing right now, and that's a good thing. But is there something, I mean, are we really giving medications to satisfy our need for a number on the monitor that is a blood pressure, or are we actually visualizing the microcirculation where maybe that much Levofed isn't a good idea? Less, accept a lower blood pressure for better tissue perfusion may be the right answer. Now you have to have the pressure head, I understand that, but there might be something else you need to do because upping the dose of Levofed is only going to make the situation worse.
And what you have now, that device allows you to do telemedicine.
Yeah, that's another good point.
You now monitor the patient in any place like ICU constantly.
Yes.
And you know that where is the patient in advance to acute situation.
Not waiting till 6 in the morning for an elevated lactate.
Exactly.
Or an elevated, something else wrong.
From that perspective, the device, I'm not trying to explain how big is the device, I'm saying that's the future
yes.
of prediction.
Yes.
What will happen to the organ and the failure. Mm-hmm. Simple as that.
Yeah, I think that's very interesting. Do we have the phone lines open? We never opened the phone lines. Yeah. Open the phone lines. And Dr. Majewski, if you could just we'll open the phone lines in case somebody wants to call in. Do we have any questions on FaceTime or whatever that place is called? Do we have the phones open, yeah?
Yes, we put them out.
Thank you. So we can keep talking for just a second. So obviously anesthesia would be interested in this device, Dr. Majewski. Cardiac surgeons would be, intensivists would be interested in this device.
Army.
Patients, the military would be interested in on this device, of course.
Well, one very important group is the newborns.
Newborns?
If you have newborns, babies that are in the in the intensive care unit, the neonatal intensive care unit, the main issue is that the capability of monitoring these small babies is very limited because they are very small. Mm-hmm. Therefore, if you can, by looking outside on the skin, and that has to be developed, to put the probe on for example, on the forehead skin, and measure the EEG together with our parameter that we tried already in babies. We can glue or hold a probe on the scalp, outside of course, not opening anything, just putting it with a very low pressure and continuously monitoring the skin. The skin will behave exactly as the urethra in terms of giving us an early warning signal what happened to the baby. Many times they have apnea event. Now you want to know what happened during the apnea and to alarm the team that this baby, no one is staying next to the baby all the time to look on the monitor. So if you have a bell that will ring you that says that there is some sort of drop in the score, the metabolic score, you run to the baby and you see what happened and take care on it.
Yes.
So it's a very important alarming system, and the market is very huge, and the need in these small babies is more significant and important as compared to adult patients because they have maybe other ways to penetrate to the body of this adult. But in the baby, you are very limited to go to, even to the blood vessel is very hard to go inside.
Mm-hmm. So I would like to, I do want to just ask you, and I thought of this here. So we have, very, I think, well established and understood vital signs, right? Heart rate, blood pressure, respiration. Would you consider this to be, this device, another vital sign?
Well, I hope that we have to prove, but I think that there is a very good potential that our index will turn to be a new vital sign that will actually will be complementary to the existing one. We are not going to replace any other methodology. The whole idea here is not to change the usual activity of monitoring,
Right.
but to add a lower level, namely lower in the tissue level of events that we want to monitor. And this, for example, you mentioned using the microcirculation inside the mouth. That's measure just one parameter, but it's the same idea.
Yes. So you have the heart rate, blood pressure, respiratory rate,
And the
and
hypoxia.
you have tissue
Oxygenation.
tissue oxygenation score, or tissue perfusion score, or whatever you want to call it, index.
Actually, tissue oxygen balance.
Tissue oxygen balance. Okay, I've got to get that. I've got to get that right. I have a question from John Ingram. Other than inserting a bladder catheter, and I think you've answered it, but maybe you can elaborate a little more. What other means are you able to monitor other tissues, such as heart, etc.? Does the probe have to be in contact with the tissue if you're trying to monitor a specific organ? That's the question.
Okay, so I will answer to the last part of the question. Yes, you have to be in contact. The probe has to touch the tissue because we are using light that has to penetrate to the tissue, and the best way and the most efficient way to get enough light into the tissue is to have a good contact. Now, I have to mention that one of the wavelengths light that we are using is a UV light for the monitoring of the mitochondria. And this penetration of the light is only efficient enough only when you touch the tissue itself. So actually you are limited. You cannot do it from outside the body. You have to touch the tissue that you want to monitor. Okay?
Unless it's a baby,
so
in which
you
case
can
the
adapt
skin
it.
is so
For example, let me just give one example, okay?
Yes.
If you have a patient that's having a kidney transplantation, okay, let's assume that we get a donation from a patient that died, and then they take the kidney out and preserve it and transport it within a few minutes or hours to the recipient patient that needs the kidney transplantation. So you need to monitor this kidney in 4 different stages before you take the kidney out to make evaluation whether the kidney is good enough to be transplanted. We can do the test using our device. Number 2, you want to measure during the transportation, okay? It may take few hours. You want to make sure that the kidney is not deteriorated during the transportation. And then once you do the surgery, by the end of the surgery, when you transplant the kidney, you want to make sure that the kidney mitochondria is functioning once you start the blood flow to the kidney after the transplantation. I was doing this kind of measurement in patients in Tel Aviv Medical Center years ago, and we found, for example, in one patient that they opened up the renal artery after the operation, and the tissue did not respond. I didn't see any change at the tissue level, although the large vessel was full with blood, but the microcirculation and the cellular level didn't get enough oxygen. So I told it to the surgeon, and what he did, he used some drug to take vasodilatation to the vessels, and then blood start to go to the microcirculation.
Wow.
So this is part 3. Then you can leave, in this patient, you can leave a small needle in the kidney for 24 hours and suture it to the kidney, and then it will remove automatically after 24 hours. But during the 24 hours, you can measure that the balance of oxygen to the kidney is normal and not deteriorating for some reason. So
And the
4
kidney-
stages in this procedure you can follow with our device.
And the kidney is pretty sensitive. The PO2.
Oh yeah,
PO2
all
is low.
organs. Now we are talking here about response time of seconds. Okay, for example, if you cut down the oxygen supply to the brain to 0, within 10 seconds or 12 seconds, you get almost 0 oxygen in the brain.
Wow.
So
Wow.
it's a term of seconds. Now, of course, if it takes 2 minutes, 4 or 5 minutes, you start to get damage. If it's more than 10 minutes, it can be irreversible damage sometimes.
Yes. Yeah. Well, probably more likely than not. I could probably go a long time. I probably could go 30 or 40 minutes, because I have a very
you can go 5 hours.
I have a very small brain.
We just scrapped the surface here.
Yeah, we have. That's a very interesting concept. Very interesting.
You can
indeed.
do on that device, I would say, organ by organ. We have many experiments. We're talking about 50 years of experiment.
Yes.
And books and publication. We just, Avram did a review article, and the audience is, I'm happy we'll give him the reference and they can read about it.
Yeah, how can we get this information, by the way?
We will do a press release about it, about the article that was published.
I need to add that to this, though. Okay, so what I'll do is when you send me that press release, can we tag it to our video here later for people?
Yeah,
Magic?
we can do
Yeah, we are a public company, so we need to do press release on those.
Okay, very good.
We
Excellent.
are a
And then
publicly traded company.
And then, okay, so, and what's the symbol?
B like Bravo, India, X-ray, and Tango.
BIXT.
Yeah.
BIXT. Okay, very good. So we need that, and we'll add that to the tags on this video, Magic, when it's over. Listen, Dr. Majewski, I can sit and talk to you for another few hours, but in deference to you being so late, I want to thank you so very much for being kind enough to stay up with us so late. I'm going to have Dr. Platt start with the BXT25 nanoparticle presentation. If you would like to stay on, you're more than welcome to, but if you'd like to try to get some sleep, we would certainly understand that.
I will prefer to stay a little bit and listen to the discussion. Maybe I will be able to add if there are some more questions later on.
That's
about
excellent.
the drug efficacy measurement and so on.
Fantastic. We would, in fact, I do, because if you can do that, I would be so appreciative, because I want to know if you have studied. Now I want to know if you have studied the tissue oxygenation index balance, the tissue oxygenation balance with the use of the BXT25 nanoparticle. So now we're going to get into that though later. We're going to let you do your talk, and then we'll circle back with that full discussion. Please relax, and we'll pop you back on the screen after Dr. Platt is finished with his presentation. If you have anything to ask, just buzz in and we'll go to you.
Okay.
Thank you, sir.
Right.
Okay. Dr. Platt, you ready to give your talk?
Yes.
Thank you. Okay, where do we go?
Go to the next slide. Next slide. Okay.
Do I need to, do you want to sit here so you can see it on this
No,
screen?
I see it. That's fine.
Okay.
I will start talking.
Oh, you want me to go to another slide?
No, it's okay. I'll let you when I'll need the next slide.
Okay.
So-
here, you can hold it.
Okay.
Yeah, you can do it yourself.
Okay.
Have you been certified? Have you been certified?
Yeah.
Okay, we're good.
So about I would say in the 90s, early 90s, I read an article about HIF1, hypoxia-induced factor 1. And the whole idea was, okay, let's describe that. By the way, those guys who discovered it got the Nobel Prize a couple of months ago. So that's the first.
It was in the 90s, so they just won the Nobel Prize. That's a long time to wait, man. It's down. Okay, you're back up, Doc.
So the question was, for many years, how you stabilize a oxygen carrier, and there are a couple of methods. So I choose, as a carbohydrate chemist, I choose the method of using the chemistry that I did my PhD and my chemical engineering degree, and I decided to modify the hemoglobin with the sugar. So if you look on the slide, if the audience look on the slide, they can see what actually happening here. You take the red blood cells, you take out from the red blood cell the hemoglobin, and then you separate the heme out of the globin, hemoglobin, and then you replace the globin, the carbohydrate piece, you replace the protein, globin, with a carbohydrate polymer. So what you get is a heme carbohydrate, more or less. It's just, it's more complex than that. But in principle, I want the audience to understand the concept. As a consequence, you get actually a very interesting situation. You have a solution now that has a molecule that is 5,000 times smaller than red blood cells. It's stable in room temperature, okay? And it can deliver oxygen. Next slide. I'll do it here.
I noticed too, you have on here that you're free of reactive oxygen species, which is a big problem with reperfusion injury. But
for sure.
I don't mean to jump on top of your talk. I just wanted to mention it.
Yes, yes.
Because I saw it.
I may miss here and there some details because I need to go over that quite quickly.
I know, and we'll take your time. I think we have plenty of time. I do know that my colleagues, though, are very concerned about reperfusion injury. It is a big problem, whether it be on coronaries, and I forgot even to mention earlier with Dr. Majewski talking about using the probe on the heart distal to the bypasses, because we measure flows and we measure runoff, but do we really measure effectiveness? But we'll go back to that question during the discussion. I'm so sorry.
The molecule, what we're talking here is a complete control on the amount of oxygen molecule that you deliver to the tissue, basically. It's not like in blood that you don't know, because of the nature. Each red blood cell contains 300 million molecules of hemoglobin, and it's hard to calculate what is the efficacy of that delivery. Here you have a pure calculation, very accurate, how many oxygen molecule you will deliver, and you control it. There's a total control on that. So it's like a chemical reaction, basically. So when you look on the limitation of blood, if you read that, you can see that you have, for example, blood is limited.
Yeah, we're having that problem now.
Right. You need refrigeration, right? It goes bad after 30 days. And you need what you call I would say, there's a problem of the size sometimes, the way you administer that. So all those problems goes away with this type of molecule. First of all, stability. I calculate, you know what's Arrhenius plot? It's any drug need to be Arrhenius plot. It means you torture the molecule with heat, and you ask yourself how long the molecule is stable in a certain condition. We're talking about indefinitely.
Really?
Indefinitely. You know why? Carbohydrate. Very stable chemistry. The second thing is the refrigeration. You don't need refrigeration. You can actually boil the molecule. It still retains the ability to bind to oxygen, because the heme is a chemical structure. Remember, now you don't have a sort of biology. It's more chemical, pure chemistry. It's chemistry on chemistry. The heme bind the oxygen, and that bind to a support mechanism. In nature, the support is the protein, the globin. But here, I replace it with the sugar. And by the way, I'm the only one who did it. So we're talking about totally new approach to the stabilization of those type of molecules. People try to do it for almost 100 years. I'm not the first one. And people were quite successful to stabilize with the chemistry. I'm not the one who established the chemistry of the stabilization. My contribution to the field is the carbohydrate stability or chemistry, which was not there before I play with that.
Mm-hmm.
That's the story. So if you look on the next, I will do the next slide. The next slide describes the size, right? We're talking about red blood cells compared to that molecule. The amount of, the ability to carry oxygen has also a size solution. What you do is that you reduce the size, you create a lot of benefits. For example, you can create a capability of oxygenate tissue in a completely different configuration. The carrier, in this case, allow you to create a different curve of saturation. It's not anymore what you know is blood. It's not blood. It's not artificial blood. This is an oxygen. It's a synthetic oxygenation molecule, which has a completely different behavior than blood. It's not blood.
See, I'm familiar with Perflubron and also Fluisol.
Yes,
I remember those.
Those products, they are not our competitor. Okay, they're simply molecules that were developed for many years, but they were not successful getting into the marketplace.
Why?
For different reasons, stability, and also the ability to deliver oxygen. I mean, people doing experiments show that it delivers oxygen. I believe it's a philosophy. It's a philosophy of chemistry. You could say, how come that chemistry is, you can get philosophy in chemistry, but I believe that if nature choose heme as a way to deliver oxygen, chemists can come and say, you know, I can do it differently. I don't want to compete with billions of years of how oxygen is connected to a carrier. All I want to do is to control my capability to carry the carrier. But I don't want to compete with the carrier. That already, nature already solved the problem with the heme. It's conservative structure. And even today, people really don't, chemists not fully understand all the, I would say, I call it a secret, that heme contain within the ability to carry and release oxygen. So if I have a capability to stabilize something that was studied by nature for billions of years, I don't want to compete with that. And that's the reason they had problem.
Yeah, I know they really weren't. I remember it was a hot topic for a very long time, and then over the years it has just sort of dropped off, and I really don't hear a whole lot more
Efficacy and
about
toxicity.
it. But you bring up a very good point, and I don't mean to interrupt your flow with your presentation, but with what's going on with COVID-19 right now, we have a lot of people, blood banks are scrambling. They can't keep up with, and the need is still there. People still need blood. The blood that they have is going to expire. We do have a problem, and we need to, and our blood availability in blood banks and our use is a very narrow margin. We really don't have a surplus of blood and can exhaust blood banks very easily. And this current problem is one example of it. Finding an alternative to this is going to be very important. Please forgive me.
That's one of the drugs that is in development for many years, the fluorocarbons. So people try to do all kinds of, I would say, they try to do encapsulation, all kinds of method of delivery. I'm not sure that, I'm not here to tell the audience that that's not the right technology. I'm going back to my philosophical chemical view that there are things that I don't know, we don't know about oxygen delivery, which I don't want to explore and find out later what's happening with that chemistry that has a problem. From time to time, company try to do it, that doesn't work. Not enough data, a problem with toxicity, certain patients, certain disease indication. I think that the way to go is to make the natural heme and try to stabilize in an intelligent way. That's my view. So I'm
and it
just
makes sense. It's they're both are naturally occurring substances, readily already existing in our in our physiology.
By the way, the sugar that I choose to play with in the chemistry, it's over the years I specialized in neutral sugars. Sugars there are three sort of kingdoms of sugar. There are neutral sugars, no charge. There are acidic sugars, and there are basic sugar. And since I'm not a neutral, I'm not a basic sugar, I'm not, it's like a different area of expertise. I concentrate on neutral sugars. The reason I like them, I think that they are the most suitable sugars in many ways, not always, but many ways, they don't have charge, so they are invisible to the immune system. They actually are not recognized by biological system because they are neutral.
So essentially they're inert.
Yeah. It's hard to play with them and to do the chemistry that took me years to understand how to manipulate and do the chemistry. But in the end of the day, the beauty, they don't have charge. So they don't bind to anything. It's like Teflon. So I actually put Teflon chemistry with the heme.
Teflon, we might want to use something else besides Teflon. Teflon had a bad reputation.
I'm talking about nothing is stick to it.
Let's use the new stuff, the copper chef.
Okay.
Yeah.
But
the granite stone. Yeah.
Okay.
Nothing sticks.
Maybe the analogy is not the right way, but I try to say nothing stick to it. Okay.
Yes.
Okay. So the next slide will give you the advantage of what you call the oxygen carrier, and I will say go down to the most important thing. It's just non-toxic. I want to say that's now people try to do different modification over the years. We need to do ours. We need to go to healthy volunteers and show that it's non-toxic. But we didn't see, to date, in animals, I didn't see any toxicity.
Well, blood's not universally compatible. You can give ABO mismatch is a huge problem.
Sure.
Antibodies a huge problem.
For sure. Each one of those points are valid, but I wanted to concentrate on that thing because people say, what is and obviously no immunosuppression. It's very important. So we're talking about bioxytran combine the heme with a copolymer. That's the concept. That's what we are as a company, what we're trying to do. The implication of that is that it's unlimited from that perspective. We're talking about an oxygenation molecule that has a standing alone, I would say, duty to perform delivery of oxygen. So One experiment is delivered to the brain. You can see here in model in mice. In case of, it's not validated. It's very difficult to do those models. You see there's an oxygenation after clumping the blood to the brain. I will not say that the best experiment, but we tried that one, and I will go back to it. But that experiment, all those experiments was done independently. We were not involved in them. Over the years, the company is new, but they will use the similar product to that, and similar to the BXT25, which is the independents actually show how great you can go with that and how strong you can show that in animals, for example, it was replaced their blood. It's published, and that experiment demonstrate, again, was done independently, injecting the similar products, and replaced the blood of those dogs, and they recover nicely. So people did it over the years. It's not the first time. But that direction, that you can make what I called oxygen material, is valid. It's not a dream. It actually can happen. About a couple of years ago, one of the problems that, remember, I'm doing that the audience need to remember that I'm doing it for many years. And each time I try to solve another problem. For example, the next problem, I start understanding that about 15 years ago, that if I do the molecule, I have maybe a major problem of not distinguishing between nitric oxide and oxygen. Because naturally, there is a regulation of the ability to distinguish, the heme can distinguish between nitric oxide and oxygen binding. If you take out the nitric oxide from the vein, you will actually increase blood pressure, so that such a product can kill a patient, increase of blood pressure. So we did experiment in Harvard Medical School independently, and we showed that type of chemistry was predominantly oxygen binding and didn't show nitric oxide scavenging. This is extremely important issue in toxicity, that you don't need to worry about increase of blood pressure. And that was done in diabetic mice. It's a known model that you inject and you compare it to other similar products, and then you measure the blood pressure. If it doesn't go up, the chemistry is okay. So that's a big thing that I work on that for a long time. How to create what I call predominantly oxygen binding to the heme and not to compete with nitric oxide. Mm-hmm. Very important point. So that's the animal experiment. I describe here the animal experiment. I can send the people the slide if they are interested. I don't want to get into it. I just put it there. I think that's important for them to summarize. If they are interested in the subject, they can go to the slide and really I extract the main point. The main point, the different groups, was done very nice, very correct, with positive and negative control on the experiment. So the question is that what is the value of the molecule? Now, the parameter that were there for years in another product, we do a similar product to that existing product that was approved for animals health, for giving them a blood substitute. So we have a similar, I would say, parameter. So there's nothing unique about that. When you, what you call artificial molecule, it's basically the same. It was done, approved by the FDA. That was for animal from another company. So we do something very similar. I'll just give you I give here the main parameter that it's not like we have and there's suddenly a viscosity goes 5-fold because we did this with carbohydrate. The parameters are the same. So it's not like we're expecting any changes from what was done to date. The carbohydrate gives us stability, as I said, and the ability to manufacture that in a very precise way. But it doesn't change the basic parameter that was before the FDA set up with other similar products from the description what it need to be viscosity to a range, and it will be need to be a coefficient will be in that range, and you understand it's there. We are the same standard material.
It's a small molecule.
It's a very small molecule, 6 micron.
Yeah, it's small.
So that described basically the experiment with the nitric oxide scavenging. What we have here is two groups. It looks like the graphs are kind of condensed, but it is what it is. That's the way people do the experiment in those publications. I didn't do it. It was done by third party, which is good. Mm-hmm. And I would say I can refer people to the articles. If they look into it, if they want to understand it fully, there were two groups, three groups, control. The control was unknown, was not given to the researcher. They didn't know what's A and B. And we see a very distinctive difference in the increase of blood pressure between the other product and our product. So we basically, I would say, solved the problem. That's the same experiment. In this case, it was done with a different product. We compare it to a list of products, and we published that and demonstrated in a conference a couple of years back, and described that revolutionary capability of not having nitric oxide scavenging, which was the basic or the reason why carbohydrate allow that nitric oxide scavenging from happening. Okay, so the carbohydrate is the key why the oxygen is attracted to the heme and not nitric oxide. What are the implications of such a product? I want to look on this slide. This is a very important slide based on reference on the top, on the bottom. And you can see what it means to have an oxygenation molecule. And I want you to look on the left side and what it said, aging. Aging is a function of hypoxia. People today going to hyperbaric chamber to get oxygen, to revitalize their tissue. It's in dementia, it's in skin rejuvenation, and the other disease. Every disease that you can think about, hypoxia is involved.
An element.
Yeah,
of it.
Very important. Recently, to give you an idea, it was an article by University of Louisiana State University that showed that no drug is approved for Alzheimer's disease. They showed that after 66 times, when they treat patients with hyperbaric chamber, which is totally impractical, they took patients and they show with a PET scan a major improvement in their disease. Actually, it blocked the progression of the disease. And actually, in two patients, It's published. And we're talking about hyperbaric chamber. We can do the same thing 20, 30 times more efficient and much more powerful by injecting them with a molecule that can oxygenate their tissue and their brain.
And what's so important, and I hope you don't mind that I ask questions while you're going through the flow of this,
not
but
at all.
you know, one of the things that's fascinating about this is that being that small, you're no longer depending, I'm assuming, and this is an assumption, that you're not depending on the microcirculation anymore.
You're not depending, completely independent.
Once it gets in, it's going to diffuse, it's going to be in the lymphatic system, it's going to diffuse through the tissue itself.
Yes.
Because it's so small.
Yes.
Hmm.
So, why we choose stroke? As a small company with a drug that it's, you know, new, what you do, and I think that's our medical director. I have two of them, very experienced. We think that we can put a major impact as a proof of concept, show that how powerful the technology to help stroke patients, because they have a period of about what you call the golden hours, that they don't get anything. And the reason is that you don't know if it's hemorrhagic or ischemic. You don't know if it's a blockage or blood is flowing all over the place.
You have to get them scanned. You got to get a CT.
And they for 2.5 hours, 3 hours, sometimes 4 hours, sometimes more, they don't get anything. Try to hold your breath for 4 hours. See what happened to you. Mm-hmm. You die. Mm-hmm. And that's exactly what happened to the brain.
That's why they have deficits.
That's what happened to the brain. Mm-hmm. So we said...
Only shorter than a few hours.
For sure. Mm-hmm. So we said, what happened if we inject that molecule right in the ambulance or in that time frame before any MRI and treatment? We will get oxygen to the part of the brain that is blocked. Why? Because the molecule is so small, it penetrates through the clot. Mm-hmm. The clot is like a fishnet, and the blood and the red blood cells get stuck behind the fishnet. But we're talking about a molecule that is 5,000 times smaller than red blood cell. It goes through because the size of the fishnet is big enough for that molecule to go through with oxygen in it and release the oxygen in that part of the brain.
Now, what if it's hemorrhagic?
We don't care.
So you don't have any blockage there. It's just going to come out. It's going to leak out.
Yeah. That's it.
Now, the damage
so we don't
on the tissue may not be reversed, because that's, you know, the blood on the brain is not.
But the idea is that you don't care anymore to distinguish between one and another.
It's true. Your treatment will be the same.
Yeah.
Because this has no additional, it's not going to do any harm to the tissues that the blood, if it is hemorrhagic, is not already going to do.
Exactly.
And you still will, perhaps, distally, I'm assuming from the hemorrhagic area, be able to get oxygen to diffuse across.
Correct.
Possibly.
And that's going in circulation for 9-10 hours.
Oh, wow.
The 10 hours, half
so
time.
So it's replenishable.
Yeah.
By the lungs.
Yeah. We go in circulation for 10 hours. Half-life 10 hours. 9-10 hours.
Mm-hmm. Interesting.
So those are what I call the companies in the field. Just if you look on the color, just look on the color on the bottom. We cover many area in stroke that the others cannot cover. And the only reason is because we're talking about, I attribute that to the carrier, to the philosophy. Remember, heme conservative. The sugar is not with a charge, just a supportive mechanism. In the hemoglobin is the same. Globin is the supportive mechanism to the four heme sugar, the heme structure that carry the oxygen. Only there are four heme of every hemoglobin, and we just replaced the globin, the support mechanism.
Your scaffold.
So the outcome is you can actually show in any area you have a sort of benefit. Okay, so and I don't want to get into it because we don't really have the time too much, but that slide is a discussion of like maybe half an hour. It's
Let's
very
discuss
loaded.
it.
What?
How many more? I mean, if you don't have that, but let's discuss it.
Eh,
There's a lot. I mean, let's unpack it.
Yes, I'll prefer to go to another slide.
Okay.
That slide is, I would say, it related to if you look on other companies, what they do, I think that each one will have a place. It's not like they are inferior or superior, or if one of the companies will get it approved, they will have a place. So I really think that all of them should have a place. I think that in our case, our contribution, that molecule contribution, like any other drug, you know, I tell you, for example, aspirin. I mean, who knew when 100 years ago that aspirin would be good for stroke or things like that? You follow me?
Or heart attack.
So we don't know. Once the drug is approved with the parameters by the company, we never know what the benefit. Because you can have a drug there that maybe it didn't work so well in stroke, but it can shrink tumors for some reason.
I have to be blunt. You know, I don't think when I first heard that the bioxytran could perfuse clot, I was I'm just to be blunt. I was like, okay, you know, let me hear this. But apparently, it's not the only one.
Yeah, not the only one.
So, perfusing clot is actually something that can happen. I really have to say I was very skeptical.
Also, the others, what they do, for example, a company Novox Pharma, which I know very well in Arizona, they do what you call more scavenging. They actually take the oxygen from the surrounding and they deliver that. There are different methods of how you play with the oxygen. For us, the oxygen is like hemoglobin. You need to go to the lung to pick the oxygen. Some companies, there are chemistry that you inject the chemistry, and the chemistry that you inject scavenge the oxygen from the surrounding tissue. So it's kind of it's a different method. I'm not saying it's good, it's bad, it's just different.
It's just different.
Different, right.
Fair enough.
Okay, so
Good. We're getting into the pandemic.
yeah, we're going to what happened recently as we looking on what's going on around us and understanding the COVID-19, it's very clear that patients get in the situation of ARDS. So, number one, the question is, why suddenly a company that dealing with what I called oxygen or artificial oxygen delivery, right, suddenly dealing with COVID-19. So what happened? And the story is that it related to the way we wanted in our stroke trial about a couple of years ago, 4 years ago, the idea was very simple. If I want to go to the FDA and I want to show to the FDA that by giving
He has to stream it.
maybe I say something?
It was your fault.
We back.
Okay, yeah, we have a lot of people watching and they're telling me the stream went down. I know, we fixed it. Okay, good. Okay, are we good? We're back?
Yep.
Welcome back. I'm sorry, everybody. Please forgive me. We, for whatever reason, are having a heat problem in the box. So they're going to fix that after the program today. So I apologize for the technical difficulties. Doctor, please continue. I'm sorry.
So what a company like us suddenly doing in coronavirus? What do we have to do with coronavirus? And the story is that for the stroke, I was when I start dealing with developing the drug for the stroke, one of the problem was okay, if I need to develop the drug, I need to show that I have benefit, medical benefit. But for that, there is what you call NIH stroke score.
Mm-hmm. Familiar with it?
Very cumbersome, almost unscientific method that require many patients and very expensive. And actually prone to fail because you maybe need 5,000 patients to show that you have a benefit, medical benefit, with the delivering oxygen. We're talking, remember, we're talking only maybe period of 20 minutes, half an hour, maybe hour and a half, maybe 2 hours. But still, it's a very small window compared to RTPA treatment or lumpectomy that you need to take out the clot. So what is the benefit that I'm giving the patients? And the most important thing is I have an oxygenation material. How I know that I oxygenate the tissue? And that's how Avram Majewski, Professor Majewski came to the picture, and then how we actually developed the technology that will have the ability, FDA approved. Now we have a technology that say the tissue is oxygenated. So now I have two things. I have a drug that oxygenate the tissue, not the pipes, the tissue. And I have a device approved by the FDA who's saying, yes, the oxygen is there, and I know how much oxygen I give to the tissue.
But that's going to require, obviously, well, in real time on any one patient, that would require a craniotomy. So you just have to validate it, that it actually occurs in animal studies, I'm assuming.
Yeah, animals and human. And since the device was approved in ARDS patients in the ICU, our idea was, okay, we're doing healthy volunteers with the drug, and then we do with the device ARDS, we show a hypoxia, and we're going to stroke patients as an intermediate for functionality. So I can get attraction by the drug company to do a collaboration. Because now I show a hypoxia with a FDA-approved device.
Now I'm going to throw something out at you.
But you see the clinical development. That was the plan.
That makes perfectly good sense to me, but I'm going to throw a little, I'm just going to try to understand. Your device, your drug, we're talking specifically about the Bioxytran,
BXT-25.
The BXT-25. When I'm assuming it's oxygenated. Is it carrying oxygen in the delivery vessel itself? In other words, the bag. Is it already oxygenated? No. So it has to go through
IV.
Right. So
go to the lung.
Right.
Pick an oxygen and like any red blood cells.
Right. So in ARDS, if the lungs are not functioning, how do we get the oxygen on the molecule besides a mechanical device?
Or you put it with the machine.
Could you, just out of curiosity, and again I'm just thinking outside the box, I want try to remember me if this works, but could you run it through an oxygenator first before adding it? Now, that would get a first pass oxygenation, but once it got out to the tissue and returned to the circulation, if the lungs aren't working,
you can do whatever you
can't
want.
replenish it.
You can do whatever you want. You can do it. No
I'm just thinking about the drug itself. To infuse it first pass would give you oxygenation before it got to the lungs.
You can do that.
But it may not benefit because it's just going to be one pass. It's all going to get used up. That was a good idea until I thought about it. But go ahead. I probably shouldn't have said it. Please forgive me. Go ahead.
Anyway, so the question was, okay, so we're doing ARDS and we have a device approved that we know that there's a hyperoxia. And then the coronavirus came to play in the last few weeks. We said, wait a minute, we have actually a drug that we can give patients before they fall into a very difficult way.
Ah, yes.
We actually supplement them, not with that device that goes into here.
Or a BiPAP.
Or we give them oxygen, we supply them hyperoxia in their system. Maybe they will not get difficult.
Because they're going to have much more oxygen carrying
capacity. Exactly.
Okay, well that makes sense.
So, or I can go to the most difficult patient and still can help them because the molecule is so small. It can pass the liquid in the alveoli.
Ah.
Because it's not red blood cell. Remember the story about the stroke? I have an advantage of the size.
Yeah. Yeah, less than 500 kilodaltons.
So that's why we said we can actually take that drug for coronavirus patients. Besides the fact that we can supply unlimited amount of material. You don't need blood donation or something.
Have you discussed this with anybody at the CDC or FDA or NIH
we have
or somebody?
We have early discussion to get a grant from the, like DARPA and we are in the early stage. Everything happened the last few weeks, so
yes.
we're trying to, it's not a normal drug development. We need to push it and we need money. If we have more money, we can push it very quickly.
Well, the safety is there.
Yeah.
You're talking about carbohydrate and heme. You're talking about two things that already exist. So I think that would pass muster very quickly.
It's very.
But I would think that the vice president's task force on compassionate use, on the coronavirus task force, being that this is so safe, would just release it as an option and see. I mean, this is really a game changer.
It's a game changer. Absolutely.
By the way, I got a message from somebody who said amazing. I have a lot of questions coming over. So I don't know how much longer you have to go.
I'll go because I have more slides I want to explain.
Yeah, let me. So everybody hold off on your questions. I've got a lot of them from a lot of people. Just be patient, and I promise I will get to all these
That's the
questions.
headline on the ARDS that's going on today, okay?
Cytosorbents. Somebody just asked me a question about it. I just got asked a question about cytosorbents. So if you're listening, let me see who it was. I can't remember who it was. It was somebody in Germany.
Yeah, that's what I said
Yeah.
in the text message.
Yes, so Hama on Twitter, here's your answer.
Okay, so you can use the ECMO situation because now you have. By the way, in the whole discussion, I didn't discuss organ preservation, organ transplantation. Remember what's the problem? You have an organ. You want to transfer the organ. There are methods of solution, like cooling that. We're talking about now room temperature.
Cardioplegia.
Now you have talking
Perfusion
about
devices.
Exactly. Now you're talking about a very powerful solution in room temperature. That it's a small molecule who can actually preserve the organ much longer.
Especially hearts, because you have
Especially
4
heart.
hours.
Maybe you will get it for 2 weeks. You think I'm exaggerating?
I don't know.
No,
I don't
I'm
know.
not exaggerating.
You're a scary professor to be, so
I'm not exaggerating.
I don't know.
The solution.
I was pretty skeptical about the perfusing clot, so, you know, I was wrong about that. Probably wrong about this too.
The idea of organ preservation, organ transplantation, and monitoring that, as Professor Majewski described, that combination, that you know how much oxygen goes in, you know that you have the ability to give it all the way in a very calculated situation, you actually prevent damage, hyperperfusion.
Yeah, that makes sense.
That's it.
Just
So you have, for the first time, it's not just throwing the organ into a box and say, let's give it a solution to oxygenate. You have a monitoring on the oxygenation of the tissue. How many molecules of oxygen are delivered in every single millisecond to every cell with the device, and in the same time you control stoichiometrically how much oxygen goes into that cell. That's the control. That's how you can keep the organ alive. Room temperature.
This is incredible stuff.
So
Incredible stuff.
when I put in there, I put that, by the way, what about trauma?
Yeah. And
We forgot trauma.
Yeah, massive trauma with cardiac arrest. Because right now, what are they doing? They're doing
What about
profound hypothermia, freezing them.
What about anemia?
Yeah.
What about ischemia in general?
What about Jehovah's Witnesses?
That's of course.
Could need major surgery, but won't take blood.
What about wound healing?
Yeah, big problem. What is that? It's all secondary to oxygenation.
You want to tell you?
Sepsis.
Sepsis. I'll give you another idea. What about cancer?
Tell me about cancer.
I did experiment that cancer is always hypoxic. They grow slow. Anything you will give to the cancer doesn't work. It doesn't divide. I gave that solution. Cancer start to divide. You give chemotherapy, cancer gone. I have experimented in animals. I mean, you overcome the basic problem in hypoxia in cancer. We're talking about every cancer. I'm not talking about just single. This is a solution of hypoxia. And I didn't talk about aging. And I know I just mentioning hyperbaric chamber replacement, which I'm talking about, you know, damage to the brain, TBI, damage from stroke. So this is what we're talking about.
Very good.
The next slide will go to which we're talking about now, concentrating on what we can do for those patients right now that are in bad shape. You need a respirator, maybe give him that solution and avoid them going into respirator. If we save, let's say 50% of the patient from going on respirator.
Ventilators.
Ventilators, they will be okay. Maybe it will be okay. We go through that crisis.
Or delay them if
delay.
they even did need it at all.
Exactly.
Depending on how much the disease affects the lungs.
Or save their life. What do you give those patients?
We don't give them anything. All we do is give them supportive care. We have the hydroxychloroquine idea. We have the remdesivir idea. We've got some other drugs, antivirals, that we're trying to experiment with and try to fast track. But really what we do with anybody, even with ARDS, secondary to influenza A or H1N1 or whatever the cause may be, is supportive care. That's all we do, period.
That's my point.
We do nothing. You know, we try to improve oxygenation because we use ECMO, we use an external source to oxygenate. But that can be problematic also when you have high cardiac output and the limitations of a mechanical device that can only treat 40 or 50%, 60% maybe, so you're getting very mixed blood that is not being oxygenated with that is being oxygenated. And depending on how your cannulation is, you could have differential hypoxemia. You could have a lot of problems. With somebody who's a very big patient with a high native cardiac output, VV ECMO as a single cannula in out is very hard to get them sufficient oxygen. Very hard. We help them, but we don't help them well enough.
So now you understand why suddenly we are in COVID-19. It's
Yes.
nothing to do, we didn't plan that. Simply part of our clinical plan end up with maybe helping those patients. And when I go further, we have here a cartoon that describes very easily to the audience how that molecule can overcome the barrier of the fluid, even debris on the alveoli.
Even with a patient that has wet
Because
lungs.
of the because
Wet, stiff lungs.
because the molecule is so small. It's not the support mechanism is the size. In this case, plus I'm not talking about the carrying the oxygen. So you can have different way to carry oxygen, but the size and the nature of the support for the carrier, which in this case is the heme, the natural heme, allow you to play in that zone that red blood cells are totally limited. They are not get oxygen.
No, your alveolar capillary wall separation exists, and if it breaks down, you've got a bigger problem. You have a hemorrhagic lung, or you have some other problem, with pulmonary edema, plasma leaking through. It doesn't work very well. You have to have that alveolar capillary membrane separation.
Now remember that
the only option.
We need to do obviously the trial. We need to get it in safety. The plan is very simple. Once we have the money, we just go and do it quickly as possible. You put it in healthy people. You measure the toxicity in healthy volunteers. Once we have that established, we go with the device that we have, and we can measure functionality of the drug. It's not medical claim on ARDS. It's taking the ARDS patient and show the functionality. If we help them, and I think we'll help them, it will end up with like another 200 patients will be a medical claim. But it's not what we're trying to do. We're trying to get to the point that as a small company, a drug company comes in and say, okay, let's put on this company $200 million and 5,000 patients and do NIH stroke score and show that the brain get oxygenated. So for us, the ARDS is just on the way to the stroke. Right. It's not the goal. If we help the corona,
happened.
Exactly.
So your track was this, and this train decided to cross your track.
Exactly.
The COVID-19 train.
I didn't finish yet the whole story because we're going from one to another, and that's by the way, I was influenced by those three guys in the 90s that got the Nobel Prize, and that's I have a question. What is hypoxia? From the 90s, I always said hypoxia, it's a disease. That recognition of the Nobel Prize, it's looking on that hypoxia not as something that you lack oxygen. That's the disease. It's like cancer. You treat hypoxia. We have a drug to treat hypoxia. It's a disease like treating cancer. That's the mistake. Everybody look on oxygenation as another perfusion thing. This is not another perfusion thing. That's so basic. It's like you're treating cancer. But it's broad. It's huge. But it's still a disease indication. Hypoxia, it's a disease. You eventually become old because of hypoxia.
Or have apoptosis.
Or have apoptosis or other things, including cancer, because cancer works in hypoxic condition. That's why you have cancer. If not, the immune system will take it out. You followed the audience need to understand it. Why I'm so excited about these three guys. They validate my thinking and my activity as a chemist to do it in terms of making the drug or the device.
Recognizing that
Recognize that
hypoxia
we
is
became a
disease
leader
state,
because
as you're
of
saying.
them. We became suddenly a leader in the field. Before that, I was marginalized. You measure oxygen, who cares? We can measure oxygen in the artery, periphery oxygen, mean nothing. All these machines that people measure their oxygen, it's not worth it because they don't measure the tissue. It's worthless. Now I said, how you can say on industry of $100 billion worthless? Yeah, that's a fact. You don't measure oxygen. You measure oxygen in the pipes. What it helps you? You don't know if the organ is dying. What is the information you have? You have a perfect oxygen perfect oxygen situation in your finger or in your arteries and all that, and in your kidneys, or you have cancer. You don't know anything. You follow my
Yes, I do.
thinking.
I will tell you this, it's just an aside. Man, we have a lot of questions. I promise I'm going to get to them. And that is that, oh my God, I just lost, I looked, saw all the questions, and then forgot my doggone question. Go ahead, let me let you talk.
That, by the way, I'm describing here the discovery by the late Nobel Prize, by Amgen, by the way. What is Amgen? How Amgen started as a biotech company? They just took the EPO, which is one of the protein that are produced along the way that's supposed to activate the red blood cells to make more red blood cells, because that's the HIF activate the cascade, then that the EPO will generate more red blood cells to get more oxygen. It's simply the same thing. My thinking was, why to go all the way to the
enzymatic protein process.
To the EPO, why not to supply the oxygen in the first place and prevent the HIF?
Yeah.
I mean, the beginning.
Wow.
What we are doing is whatever Amgen do with the EPO in the cascade of event to get more oxygen to the tissue, I say, wait a minute, I have a chemistry, I will deliver to the tissue the oxygen. Why I need to go all the way to I'm not saying
that takes time. That's a process.
It
you don't
takes
just
time.
give a shot of EPO,
Exactly.
and they suddenly, when you turn around after 15 minutes, they have.
But we can give a shot and we're okay. Boom.
Because you're actually giving an oxygen carrying molecule.
Giving the oxygen.
You're skipping all of the steps of
I'm going
stimulation to bone marrow
the
production.
HIF.
And then what do you do when somebody can't produce them?
The factor is could produce because of hypoxia.
Right.
You eliminate hypoxia, there's no HIF. So the cascade doesn't exist.
Now, can you block the stimulant? Well, of course, it's short 9 hours. You're just treating a situation.
I treat
at
the
some
situation.
point,
I
you
don't
need
let
to
it
fix your. At some point, it needs to be repaired.
For sure.
You're buying time.
I'm buying time, exactly.
Buying
Nothing
time.
more than buying time.
Buying time, yeah.
And
that
by the
makes
way,
sense.
I'm not saying that I'm the most efficient one, and I'm not saying that that's the ultimate solution. Don't take me wrong. There's no silver bullet in biology. There's always partial solution. Everyone that in the field will have a contribution.
Yeah.
We're talking about, what we're talking about? 50 billion a year, maybe in sales, $100 billion a year of sales of this type of product. So what? So it will be $15 billion in sales a year. And there will be another company who will do another 15 billion. But it's the size of the market is the hypoxia market. That's what I call it, the hypoxia, the lack of oxygen. When each one will find a way
to work the best in certain circumstances.
For that specific chemistry.
Yeah.
I'm not saying that we are the best.
Or that
I'm not saying
specific
that we are
anatomy and physiology.
Exactly. And I don't want, people try me, all the time people say to me, hey, as a chemist, you need to compare yourself to this, you are better than, superior, inferior. That's not true. And there doesn't exist in drug development superior or inferior. Every chemistry that was measured, the toxicity is known, has a place. Any chemistry. As
Very
long
good.
as you understand the...
I appreciate that. I think that's very, very, very nice of you. I think that's very good. I'm glad you said that. Okay,
So
do you have more slides? Yeah.
I'm trying here to talk about the strategy of the commercialization, which, as I said, I'm repeating why the device is important for the drug, why the drug is important for the device. They're coming hand in hand. It doesn't mean that it's a drug device approval. It means that I use the device to validate the drug.
Makes sense.
That's it. But the device has its own space. It's nothing to do with the drug. Okay? Also, we have because of the device, we have something that all the companies in this space don't have. We can pursue medical claim by the FDA. If I'll be successful, I don't know. I need to be there to tell you if I'll be able to show to the FDA and then extract that type of a claim, which doesn't exist actually. But I have the ability to do that. Remember, the device is approved. If the drug shows that it increases the oxygen in the tissue, and I don't make any medical claim on that, maybe after X amount of patients, statistically, they will agree, or maybe not. But at least I will get a drug company on board, or partner.
And that's what you need.
And that's what I need. I need a big partner.
Yeah.
Big companies will not do anything with you if you don't get to a certain level of what I call proof of concept.
Well, yes, I understand. I'm very familiar with proof of concept. And I'm really hopeful that, and we're going to go over this after we're going to, whenever you're ready, we're going to loop Dr. Majewski back in and have like an open discussion about some of these things. I've got a ton of questions. And then we're going to take a short break and come back and talk to people who stay online that may be connected. I'm hoping that people out there that are listening to this will try to keep discussing it, share this, and bring an awareness to what it is we're talking about here, because it is revolutionary. This is highly provocative, very revolutionary thinking. And it's way outside of what we're all familiar with. So that's what I'm hoping from our audience. But then the other audience that once we take our break and we're done with the CME portion of this, or the CEU portion of this, we can be more free to talk a little bit more about that and bring Mike in as well. Okay. So whenever I'll let you finish up your slides and then we'll go to these questions.
I'll finish the presentation by saying, from a commercial perspective, when you look on the chemistry and all that, the applications are enormous. So you do the chemistry, you get it, that you show that it's safe, you make a proof of concept, and then the company, you cannot go everywhere. So the company, what we'll do as a company, we will do licensing. And that's how we develop in the different areas. You can capture in very short period of time, you do 10, 15 licenses. And that's how you create your footprint in different area in medicine that you cannot be as a organization unless you raise, $500 million.
Unless you're Eli Lilly.
Yeah, you cannot do that. So or you go with a large drug company, or you go with 20 drug companies, and each one specialized in hypoxia in kidney, liver,
stroke,
acute
coronary syndrome,
any heart issues,
cancer,
anemia, cancer, and that's the idea. Because if you cannot capture.
And that's a huge thing. I did not know that cancer was that it depended on hypoxia. I didn't know that.
Of course.
I didn't know
the
that.
number one. That's number one why cancer drug fail, the hypoxia. It doesn't matter if it's genetic, immunogenic, immune method, or chemistry, radiation. It's all hypoxia.
Interesting. That is very interesting. Okay, you ready for some questions?
Yes.
Okay. So let's start up here. Can this drug will have to help reduce? Okay, can this drug help to reduce the reperfusion injury seen after ischemia, such as you see with coronary bypass surgery?
Yes.
So, how would it do that? I can tell you what my theory is, but you tell me.
Say your theory.
My theory is you infuse this via the cardioplegia into the heart so it never becomes ischemic. You don't have blood flow, but you let it stay oxygenated.
You answer the question.
Okay, so I answered. So I've been listening to you. And
by the way,
I've been listening to Dr. Majewski too. Welcome back, Doc.
By the way, we need to remember to the audience, they need to remember I'm a chemist.
Yes.
I cannot answer medical question. I have no education as a medical doctor.
Neither do I.
No, but you need to remember, the audience need to remember that.
Yes, and they do. I think everybody understands that.
So I have a limitation in my understanding in a certain level, because it's not, I don't have the, I have the education, I read, but it's not, I'll self-educate.
You don't have the clinical medical experience.
Exactly.
I understand that.
Because
we understand
in the end
that.
of the day, I'm the guy who is responsible for making the chemistry and manufacture that, but that's as far as I can go with that.
I think that's fair, and I appreciate you making that point. So the answer to your question, Amin, is yes, by avoiding the ischemia to start with. And actually, let me bring Dr. Majewski into this. So, Dr. Majewski, many times when we do coronary bypass surgery or we do peripheral vascular surgery to open up blood vessels that have been blocked by plaque or whatever the reason may be, we measure the flow in the coronary artery with a device that is it's transit time flow measurement. And so it measures the flow through the artery, the bypass vessel, but it also measures runoff. So you know if the bypass graft is actually open and has a high probability of long-term patency. However, with your device, just because you bypass a graft doesn't mean that it's a big pipe. So you can take this and look at the distribution, your device, put it literally on the heart, it's easy to sterilize, and measure the oxygen balance index and know whether or not that blood vessel is perfusing the muscle adequately.
Absolutely.
So you would also be able to use it diagnostically.
You can hear me?
I'm sorry?
Can you hear me?
Yes, we can hear you fine.
Yeah, basically you can use it intraoperatively. Once you do the bypassing, you can take a probe and put it in the distal area of the blood vessel that you treat. And you just put the probe in, measure the baseline, and then occlude the vessel for 20 seconds, which doesn't make any harming to any arm, and see if the mitochondria is responding distal to the area that you do the grafting. Mm-hmm. So you can measure the tissue level oxygenation immediately after the bypassing.
Very interesting. Yeah. Now with coronaries, sometimes you don't necessarily have symptoms until you exercise. So you're using, you're making the heart work a little harder. So you may not really see. You may see a very mild deficit, if at all, in a resting state on, let's say, something with an 80% blockage. But you certainly would know whether or not that heart is getting the right. You would probably be able to tell. If it was 100% occluded, you would definitely know. Definitely know. But that's very interesting. But I think
There's another point. Look at the chemistry. I choose the heme. The heme will choose the right oxygen.
That's true. Very good point. So
I don't need to worry about the right oxygen. The heme will choose the right oxygen.
Of course.
Right? Only one.
There's only one.
You see that?
So
you understand the magnitude of that?
I do. It's profound and it's provocative.
Because I don't need to worry in my modification what oxygen I choose from different. You see, as a chemist, you have the ring of the Schrödinger equation. Allow the oxygen to sit there with 16 positions open. Okay? This mutates, you know what I'm talking about. Different oxygen species.
Yes.
Reactive oxygen. I only can bind to one oxygen, which is the right one, with my molecule. I don't worry about the other species.
So when you have oxygen radicals, you don't have to worry about them occupying this.
Exactly.
It won't do it.
Exactly.
I understand.
But
I get it.
It's not,
which is very
many people-
important when
and
you
that's
have.
why again, I'm not sure that that's why I said I don't want to say that I'm superior or inferior to the other methodology of oxygenation. I'm saying I'm conservative in my thinking philosophically. I know at least one problem I solved. I don't go to another oxygen. That's my point.
I think it's a good point. So where do you obtain the heme? I think you had that in your slides. Does it have to be obtained from humans? I would think not, but
No,
where
I
do you
take
get
it
the
from
heme?
cow. People
So it's bovine.
Yeah,
bovine
because,
heme.
because you have abundance of that.
Mammalian. Yeah. And
Yeah,
it's
a cow give you 14 liters.
yeah.
So you can create huge amount of material
From one cow.
with one cow.
You don't even
forever.
have to take all 14 liters. You could just take a few and let the cow continue to make it.
Or you have a manufacturer. You can make tons of material.
Very good. So bovine is the preferred choice, but you could do equine if you wanted to.
I can do
It's just
anything I
not as
want.
accepted. Mammalia has to be mammalian,
Mammalian.
I imagine.
Yes.
Yeah. What precipitates
By the way, in the past, FDA approved cow.
Okay.
So it's easier to go to the FDA because everything is there. There were almost 4,000 experiments on human, 4,000 human, including approval in South Africa and Russia and other countries on oxygen, on hemoglobin, modified hemoglobin from cow. So why not to take what people already did for 50 years? And that's the reason for the cow.
That makes sense. Okay, I think I know the answer to this too, and I'm going to let you answer it. What precipitates the molecule to release the oxygen to the tissue? I would imagine and not prematurely. And similarly, what part precipitates the molecule to accept oxygen back from the lung?
I think it's related to the regular duty of heme by definition. It will be less pressure, it will release.
So it's just a pressure gradient.
Yeah.
Yeah.
Nothing
If it's
more.
low tension here, high tension here, it moves that way. If
The heme is like I'm a crystallographer, okay? We can talk later about the galactine. How
we haven't even talked about
we
galactine.
didn't even talk about the galactine. We need to mention that.
Yes. Well, why don't we do that?
Okay.
So let's talk about galactine.
But that's what I said. I'm a crystallographer, and I'm not a full crystallographer. I have a publication on the subject, but I discovered in my postdoc, I was the first person who expressed the gene for lectin. That was a protein on cancer cells that recognize only the sugar galactose. And I decided to call it galactose binding lectin, or in my publication, I call it galactin. Since then, there are about 4,000 publications on galactin. And I just announced this week that I want to create, over the years I developed antagonists to galectin, galectin antagonists to block the galectin. And I manage companies in clinical trial to manipulate the galectin, the sugar, to bind to galectin. It's a sugar that it's antagonist. And one of my biggest research subject was galectin-1. So corona, on the coronavirus, the corona has galectin-1. So I have a drug, potential drug, that bind to galectin-1 and can inhibit the corona.
So the spikes,
Yeah.
the crown portion of the virus,
Yeah, drug
is
that
galectin-1.
Yeah, it's in the publication. It's all over. I didn't do it. I mean, people already prove it. They completely cleared coronavirus in animals with anti-galectin-1 drugs. But they are not good for human. They are in model. But I have the molecule to block the coronavirus. So I'm trying to get now money to do that as quick as possible to try it on patients.
I think you need to call the vice president's task force. I really do. We're going to call them for you.
Maybe.
I mean, they always take my calls.
That's a serious. That's a serious story.
It is a serious story.
Because who know that the galectin-1 is the corona target.
I didn't even know what galectin was.
It's
So I've learned a lot today.
It's there are about today. I was the first one who described the structure of galectin, and I asked early in my career when I did my PhD. I did my PhD on carbohydrate, and I was fascinated by the fact that sugar can do so many things so precisely. That it's a fine tuning of recognition. By the way, the reason that people try to block coronavirus with the antibody on the galectin, they failed because it's not really, the antibody is too big. Only sugar can do that. I think that we have a good chance to have a drug that can clear the virus from the patient right away. It's not antibody. It's not a protease inhibitor. It's not mechanistically, doesn't go into the cell interruption of the mechanism, the virus. It's just
Or to damage the RNA or DNA.
It's a simpler clearance through the liver of the virus.
Just by blocking its ability,
To recognize
blocking the key
the
that
key.
allows it in the cell.
Blocking the key to allow it to the cell. That's it.
Wow, that's incredible. A question I have here, I think I know the answer to this too, but John, I hope we answered your question. How might this work in decompression illness with scuba divers? I think that's a nitrogen, an excess of nitrogen though that's coming out. I don't think anything would work other than hyperbarism in that case. I mean, to answer your question, I think I can answer the question. Decompression sickness for scuba divers is really that nitrogen is inert, and so you absorb a lot more of it, and then when you come out of the pressure environment, that nitrogen comes out, and you can have embolic events and so forth. And the only way to clear that is to recompress the person, force the nitrogen back into solution, and then slowly clear it with 100% oxygen environment, and then bring them out of their decompression slowly. I think that's the answer to that, I'm pretty sure. I see that the new viral inhibitor is expected to restore adaptive immune system to normal function. Is there a possibility of binding to the protein spikes COVID-19 virus, thus stopping the cell entry? Well, Bill, you just answered the question with a galectin-1
And by
inhibitor.
the way, that experiment was not done by me. It was independently by groups, it's all over the place. Simply the difficulties. People on the audience need to understand. I, as a structural crystallographer and published art scientist, with my collaborators, we try to find the right key, which is the sugar, to the right lock. By sheer serendipity, because the whole idea of cancer metastasis is the recognition of the cell in the bloodstream. The cancer cells survive the immune system by creating clusters. And the clusters can be interrupted. I just published in 1993 showing that by adding a little bit, a minute amount of sugar to galectin-3 as a galectin-3 inhibitor, I was inhibiting completely metastasis in my publication in 1993. That was the first time that I could show that I can inhibit metastasis. So the coronavirus simply has, any virus, by the way, any virus, any infectious disease virus has its own galectin. Influenza has different galectin. There are 15 galectins. By sheer luck in this case, I was working on the galectin-1 and galectin-3 inhibitor. And the corona has galectin-1. And that was in phase 2, finished phase 2. There was like 200 patients. I can take a similar thing, design it very quickly, and you have an anti-corona drug, non-toxic. That's my thing that I want to push it.
I want to come back to that.
We did a press release about it.
Don't let me forget this. We need all your press releases. Dr. Majewski.
Yeah.
Someone at the Leipzig Heart Center in Germany is asking if you use this advanced monitoring, your MDX viewer, to see DO2, VO2 in combination with ECLS, so resuscitative, extracorporeal resuscitation. I am, and I just apologize for joining the conversation late, but do you have any way with your device of measuring oxygen utilization? In other words, I think you do because you're looking at mitochondrial health. So because you could have oxygen delivery but no uptake, and I've seen that happen before. And if you shift the oxyhemoglobin dissociation curve far enough, you can actually inhibit the release of the hemoglobin to the oxygen from the hemoglobin to the tissue.
Correct.
So can you address that, Dr. Majewski?
Yeah, at this point in time, I don't think that we can calculate accurately, quantitatively, so easy the relation between supply and demand. We can, in real time, we can follow it, to follow the tissue vitality or tissue metabolic score. So it's not quantitated in the same term as the guy asked you about. So I think we have to bridge somehow to get some more information from patients and try to use the classical calculations of VO2 and DO2 and see how can we fix our concept into the same frame of calculation. Mm-hmm. We didn't do it yet because we didn't do enough patients yet.
Mm-hmm. Well, if you're measuring mitochondrial, you won't have metabolism, right? So you can, so let me just try to understand. You're measuring that there's oxygen in the environment. So am I getting that right? If I were to
calculate it in the same quantity, so some sort of quantitation problem here, how to bridge between the classical calculation way and the mitochondria. Nobody really combined the mitochondrial parameter to the old kind of parameter that's calculated. So it has to be some bridging process between the two concepts.
Yes.
Because nobody really measured before mitochondria in patients. We are the only one that we got FDA-approved device to monitor mitochondrial function by NADH monitoring.
Right.
So there is no data in patient yet enough to do this kind of calculation. So I think it's something that will be developed in time. Once we get enough patients, maybe people from quantitative biology will come and do some modeling and find out sort of calculation way what will be the best equation, how to develop it.
That makes a
So
lot
it's
of that makes a lot of sense. But I think that, you know, just sort of thinking logically, if you don't have oxygen, you're going to have mitochondrial failure, and then you would see that. So you're not only
true.
so you right. So you could infer that yes, there's oxygen there, but it's not being utilized because the mitochondria is dying. You know,
Right.
if you have apoptosis, you're going to see that.
By the way, I didn't mention a new area in therapy, which many people are not aware of that. It's called mitochondrial transplantation.
Yeah, tell me about that. That's very interesting.
You can restore tissue by taking autologous mitochondria and inject that to a tissue which it's actually you do tissue repair.
It's like stem cells, only
Like stem cells, but you inject the mitochondria to replenish the tissue.
Yeah, that's a very interesting. That's
People
gonna
do
be-
that. There was
So
an
how long
article-
have you been working on this?
What?
How long have you been working on this?
On the
On this oxygen carrying fluid?
30 years.
30 years. So 30 years of your life have been spent just on this one project.
Yeah. I'm in the modification of those molecules. I have the first patent that I wrote, which, by the way, this the only patent in the field that I stabilize heme in sugar. I got it in the 2005. That's the first one. Then I have a few others that I did. I'm basically the book, Carbohydrate Drug Design. It's about carbohydrate modification. I modified chemotherapy with carbohydrate. By modifying the chemotherapy with carbohydrate, I make the chemotherapy non-toxic. And I'm using the chemotherapy, the carbohydrate like antibody. Since they recognize lectin, I can use the antibody. If I have the right key, I can attach chemistry, not antibody. It's not biology. It's not biological material. It's a chemical structure to another chemical structure, which is in this case the chemotherapy. And now the carbohydrate behave like an antibody because it recognized the galectin.
Interesting.
That's a completely different universe.
These are game changers. So, yeah, these are game changers. Okay, one last question. No, I answered it. Okay, no, I need to ask you this question. Oh, you answered it. Okay, so that was the one from Bill with the binding the COVID-19 virus. Oh, I did have one other question. What are your views on another person in Germany wanted to know your thoughts on Cytosorb for COVID-19 patients? If you have an opinion, you may not have an opinion about that, but I got asked. Now, Cytosorb is the molecular adsorption therapy, right? So it runs the plasma through a filter, a type of hemoconcentrator that has the ability to adsorb the virus or the inflammatory mediators, right? Is that what that is? That's what that is, I believe.
Yeah, something like that.
Yeah. So it's molecular adsorption therapy, or yeah.
Good luck. Every time the virus goes to, you lower the viral load, there will be more viruses coming from other cells, you know, when they explode and all that. It's a...
So you can control temporarily, but you can't. You have to really clear it completely.
You see, when I'm talking about the
Because intracellular, they're still producing duplicates, and then they're going to release.
Yeah,
So you can clear them from plasma. You can't clear them from the cell.
In my opinion, there's a phenomenon which you call cytokine storm.
Yes, we're very familiar with cytokine storms.
And guess what? How you can block cytokine storm with introducing galectin. Anti-galectin molecule. So it's not just that I will remove the virus with the liver, which, by the way, this is a different mechanism. That's the most efficient mechanism. It's not like outside the body with some devices. We're talking about the liver. They do it in a huge capability to remove, foreign structure.
Right. It's a filter.
It's a filter. It's a
yeah.
much better filter than any filter that human ever did. But
Sure.
The idea that you can block the cytokine storm, that by itself allow the immune system to operate.
Because you have to reset it. That's true. That's what happens is your pro-inflammatory mediators overwhelm your anti-inflammatory mediators, and the imbalance just gets worse and worse and worse and worse and worse. It's a big problem.
We need to also remember, the audience listen to me and say, wow, the guy really has something. But there are today about 300 different scientists. Each one propose his own universe of view how to get rid of the virus. So but I know one thing, I'm the only one with carbohydrate. So at least I have the advantage.
Well, listen, I appreciate your humility. It's very attractive and it's really appreciated. I think you've been very, very humble in your opinions. I think you've been very clear about how you feel. This has been extremely informative, beyond informative, very highly provocative, very informative, exciting. Dr. Majewski, I cannot thank you enough for staying up so late, coming to us all the way from Tel Aviv. It's so appreciated. You too have been a very humble servant of science, and we can't thank both of you enough for this terrific CEU program. This has been very important. Now, with that said, we are going to take a very short break, only about 2 or 3 minutes. How much time do you guys need? You need 8 minutes? Can you do it in 5? Sure. We're going to do 5 minutes and a 5-minute break, and we're going to come back and we're going to talk about the financial. We're going to bring Mike Seek in, the financial investment opportunities that exist. And I appreciate so much us being allowed to separate that, because you have to be very careful with CEUs and having it appear commercially biased. So we're going to take a 5-minute break, come back, bring Mike Seek in. Dr. Majewski, if you want to stay, you're welcome to do that. And then we can talk to people who want to stay and listen to ways they can reach out, get involved, invest, help with resources in terms of networking. I've already gotten 2 or 3 messages from people with ideas of other people who they know that want to be involved in this project. So let's take the 5 minutes, and we can get some more coffee if you'd like, or tea. And Dr. Majewski, please feel free to get yourself another cup of tea, and we'll be right back. So 5-minute break, everybody, okay?
Okay.
Thank you.
Dude, I got more.
Mixed cannula. Expanding performance standards. High performance cannulae for direct and femoral cannulation in minimally invasive cardiac surgery. LivaNova's complete range of mixed dedicated products allow easy and reproducible procedures. The portfolio includes the sutureless aortic valve Perceval, Memo 4D for mitral valve repair, the HeartLink system for assisting the perfusionist in applying goal-directed perfusion therapy, and a broad choice of high-performance dedicated cannulae. LivaNova's range of cannulae suit every cannulation technique for minimally invasive cardiac surgery. Venous and arterial femoral cannulation can be performed using the WRAP venous cannula and the EasyFlow Duo. For arterial direct and venous femoral cannulation, the EasyFlow aortic cannula and the WRAP are available. For venous femoral access, the LevaNova Wrap Cannula features a dual-stage configuration for optimal drainage of the inferior and superior vena cava without interfering with the right atrium. The high-flow dynamics of Wrap improves the capability of drainage with a low pressure drop. An optional Sellinger dilator kit is available to perform percutaneous insertion. The LevaNova EasyFlow Duo Cannula is designed for a safe and smooth femoral arterial cannulation. Its conical dispersion tip design allows a more gentle flow in the femoral artery, and its red cap avoids blood backflow during obturator removal. A dilator kit for percutaneous insertion is also included with the cannula. EasyFlow cannulae are designed for safe, reproducible, direct aortic cannulation and include a malleable stylet to aid cannulation insertion, facilitating bloodless introduction into the aortic arch, as well as a special conical dispersion tip for a more gentle flow in the vessel to reduce shear stress.
The point-of-care program at Huntsville Hospital is approaching its 20-year anniversary. As we began to grow as a hospital and provide more complex services to our community, the need for point-of-care testing became very clear in our organization. Many of our departments consider the EPOC device to be crucial to their ability to quickly move their patients from entry to the hospital to procedural room. It helps them monitor the patient's status during their procedures and post-procedure, so it's very integrated into our entire continuum of patient care. If you have any patient population where blood management is crucial, then a point-of-care device is very useful in those types of settings. It can provide a quick result on a very small amount of blood, and that is really important in some of our patient population.
We are starting a new process where we're going to screen our patients on the floor for sepsis. The patient can look and actually feel okay. They will show us signs and symptoms of sepsis. So what we do when they show these signs, we get a lactate level.
Lactic acid is a product of the sepsis cascade. So with sepsis, the inflammatory light switch goes on, the cascade starts churning, and unfortunately, once it starts going, we have to stop it. It will not stop on its own. The faster the lab draw, the faster the lab results, the faster you can act on it.
The process in the lab is what the process is. It just takes a certain amount of time for the instruments to run those tests.
I mean, we're talking 30 minutes even on a stat because that's the process. That's just how long it takes. There's not really anything you can do about that. For every hour you wait to give an antibiotic, the mortality for that patient increases 8%. So now that we have the handheld device, the nurse can get those results within a few minutes, and we know how to treat that patient. If a patient is going into septic shock, we do not have minutes to spare. We have got to get it done quickly. So when you get the little beep from the EPOC that says your results are in, you go with it. You don't even think twice about it. You're like, this is good. This is what I'm going to do for this patient. The next steps are clear.
I think our mortality on the initial floors dropped by almost 50% in the first 3 to 6 months. At this point, we're about hospital-wide. I think we'll be hospital-wide another 2 to 4 months. So this has become a huge program within our system.
Without the EPOC device, it would really be hard for us to get everything done in the timely manner. So it's really helped us to get our program going.
I mean, whether you're a surgeon, whether you're a medicine doctor, whether you're a dermatologist, you want to be able to do things in a timely fashion so that you can be most effective.
This really makes a difference in patient outcomes.
Like all perfusion professionals today, we are busy and frequently short-staffed. Does taking time off for a conference create a burden for your teammates? Has reimbursement for your continuing education dropped? Have you priced the cost of going to a meeting lately? Registration fees, airfare, transportation and parking, hotel, and meals out, this totals up to having an out-of-pocket expense of over $2,500. And then you hope the content is good. Perfusion Education has heard your concerns. They have a simple and seamless system that works on any platform, computer, tablet, or even your phone. You can chat during the program or even phone in and be live on the air. The content, second to none. Perfusion Education is by perfusionists and for perfusionists. Create a free account and check us out today.
Okay, and thank you, and welcome back, everybody. We had a great doc. That was a fantastic, I understand, chemistry doc, PhD doc, carbohydrate expert, along with Dr. Majewski, who was, I read his CV. Notwithstanding that you're not a medical doctor, of course, neither am I. Nevertheless, I think that most of medicine that we have, pharmacology certainly, the pharmacology of medicine comes from chemists and people like that, scientists, basic scientists. Applying it to human care, you know, that's the purview and the art of medicine. But you know, they have to rely on people like you to give them the tools to use them the way they need to use them.
It's translation.
It is.
It's translation of science.
Yes, and we all need each other. I think that's really the point, you know?
Sure.
I mean, you know, I have a role, you have a role, they have a role. We all have a role in this endeavor that we call this big giant industry we call medicine that's filled with so many different things. So anyway, it was an incredibly informative and provocative and well done set of presentations by both you and Dr. Majewski. I can't thank you enough for that. But this part of the program, as I said, is non-CEU. This is about the ability to fund, because it takes money. And you know, as you had described, you're a smaller company.
Smaller public company.
Smaller public company, right? Public. So let's start off with, again, give us your, can we put it up on the screen? Is there any way you guys can put the symbol for the company up on the screen? What is it again? I had written it down and I forgot where it is.
BIXT.
BIXT. Is there any way you can do that, Dave? Thank you while you're doing that. And then let's introduce Mike Seek. Mike, you're here. Can you hear me?
Yeah, I can hear you.
Good, excellent. So, Mike, you are the Dr. Platt is the science guy, the chemist. You are the finance guy, right?
Business development, mouthpiece. You know, I'm mainly a biotech analyst.
Biotech analyst. And I will tell you what, you're a heck of a writer. That much I can tell you. I've seen some of your press releases, and you can definitely write. You write very well. Why don't you start this thing off with kind of where you are, what the opportunities are, keeping in mind that some people may be watching this live. Some people we are going to send this portion if they, the requested, or if I ask them if they have any interest, we're going to send this portion of the presentation separate from the other one, but both presentations, both of these will be joined together, but they can listen to this separately from the science portion of it. So why don't you start this off with kind of what are you trying to accomplish here besides, getting money to continue this research?
Well, money is really the foundation, but I think what I'd like to do is we really need to get an awareness and a movement behind this. We truly have something profound here, and we need to do something about it. So I don't want to, I'm not going to sit here and say, oh, buy the stock. Look, there's people out there, you know how the capital markets work. It's very simple. If the stock price is higher, you're able to raise money, you're able to raise capital, money comes in, project gets funded, okay? So we don't need to go there and just tell you the basic economics, okay? What we should do is we should talk about the higher calling. I've known Dr. Platt, close to 20 years. He has incredible technology. I really love what he's doing with the galectins, but the galectins are truly right now they're important because we need them. We really need them, but they're a little bit they take away from the sheer beauty of the oxygen transport molecule. You see, at the top of the pyramid is oxygen. And if you can't live more than you can't hold your breath more than a minute, probably. I just want to illustrate something. The time that I've been talking, if you were holding your breath, or if you had a stroke, you just aged. I think it's about 6 months. I want you to think about that. That's the power of oxygen. So what we really need to do is we need to get this drug commercialized. Anyhow, so let's get back to coronavirus and why we're even talking about this. David had eloquently stated, hey, how do we get into this? It was because of ARDS. So ARDS is a real symptom of coronavirus, and we feel that we could treat the acute stage people. And in that process of looking at ARDS, what we did was we stumbled upon something even more profound, which was when we found these journals. And the key journal it's in the latest press release. It was a decade of structural review of the coronavirus. Well, what we found out is when we look at it, when we look at a decade of structural review, what is the most common element in that journal article? It's the galectin. Okay, so what we're doing is we're putting pieces of the puzzle together, and we're seeing it unfold in a major way. Now, I want to talk to you about something that you see almost every day from, like, for example, Dr. Fauci. he says these key words, people with underlying medical conditions. We all hear that. But what does that really mean? Can we quantify people? So yeah, we can. What we've done is we've quantified people. Oh, if you have diabetes, if you have cardiac issues, if you have cancer, okay, we put you into this bucket. I think we can do much better than that. And I'll explain why. As an analyst, what I've done is I looked at this and I said, wait a second here. Does everyone know that there's a galectin-3 test out there? There's a galectin-3 test that's approved to measure heart failure. Why aren't we using this test in coronavirus patients? Why aren't we getting this data from people right now so that what happens is when you go and, you know, we have all these test kits out there, right? So now you get, so imagine you're this person that has coronavirus. Oh, great, I tested positive for coronavirus. What's the next question you're going to ask the person? Well, how bad is it? Guess what? I think that if we start doing some data and collecting some data, we can answer the question for that person. Imagine you're a cancer patient and you go, yeah, you have cancer, and the doctor just stops there and says, well, what stage do I have, doc? You say, I don't know. That's not a good place to be in. So I want you to think about I want everyone to think about this. We can do better, and it all starts with using, maybe expanding, a test out there and checking for galectins. Now, there's something that I wrote about. I'm a blogger, and I wrote about this, and it was called. It's titled. It's on my blog, by the way, and I'm not trying to be promotional here, but it's the latest data places Merck between a rock and a hard plate, conflict between ethics and profits. Okay, what I'd like to do here is tell you a little something, and I also want to share a graphic with you, and this graphic is really just amazing here. But what happened is the Italians about a year ago, not quite a year ago, but almost a year ago to like today, it was April 1st of last year, April Fool's Day, but the Italians came out with a study. And this study was a retroactive study on lung cancer patients that took Keytruda. Now, why am I bringing this up about cancer and galectins? Because I want to show you how profound the issue is, and we could really do something here great with this program if people really heed the message. So what happened is they did a retroactive study. This study basically took patients that took PD-L1, and PD-L1 is supposed to be, and I say supposed to be, a great prognostic indicator of whether or not Keytruda is going to work. So I mean, I'm going off a tangent here, so just give me a little latitude, and I'll get to the punchline here. So what I'm going to do real quick is I'm going to share my screen real fast, if I may. We'll see if this works. Hopefully my screen is shared. Can everyone see that?
Yes.
Okay. So this is from my my blog, one of my blog postings. Anyhow, what I'd like to show you, this is a spider plot. What happens is everyone that I want to show you how good a prognostic indicator something is called galectin, and I believe if we use it in corona, we're going to have some much better results. But check this out here. The people that have high galectin, they're in red. They have high galectin expression. Their tumors were stained for galectin expression. So if you had high galectin expression, you're on here. You have a red line. If you had low or moderate galectin expression, you had a green line. What the green line, or so what you want to be is you want to be on this side here. You want to be on this side. This means tumor reduction. This side means tumor progression. And you'll see a lot of people, their tumors progressed. And what did they have? They had high galectin expression. Now, there was some statistics on here, and here's what it is. 90% of the people that had high galectin expression, 90% of these people that had a high galectin expression failed Keytruda. This is a prognostic test that people could do. We can actually check people for galectin expression before they go in, before they get treatment for Keytruda. And then here on the other side is these had negative, they had low to intermediate galectin expression. All right, so and you can see the results. Now, 100% of the people here had a reduction in tumors, had a response. That's amazing. So it just goes to show you with one of the largest drugs out there, with how good a prognostic for galectin is. There's something to galectins. All right. Now on my same blog posting, I just want to show you something here. Look at the before. I click to enlarge. Look at the before and after pictures of a person that was treated with a galectin inhibitor. Look at the before. Look at the after. You'll see the time frame. I hope everyone, again, I encourage you to go here, but I hope everyone kind of, takes this seriously. This is what happens in cancer, and it's dramatic.
Yeah,
and I don't think many people know about it.
Before you go too much farther,
I'm going to unshare my screen.
Don't. Before you go too much farther, Mike, show us the blog post again. Don't unshare it. Go to the title so people can see it, and if they have an interest, they can go look it up. But we have to see it first. There you go. Michael Seek's blog, latest data places Merck between a rock and a hard place, conflict between ethics and profits. Okay, very good. So
and you'll see that was right done there on May 2nd, 2019.
Very good. Okay, good. People can now see that and they can find it. Thank you.
Yeah. So I wanted to do that because that just highlights the. Let me unshare this here. Give me a second. All right. So what I wanted to do is I wanted to go through this and basically show everyone. Look, galectins clearly have a place. They clearly have a place in one of the largest drugs out there. All those people that are failing, and it was around 40% of those people that fail. I don't know, what could we do with it? Maybe give them a galectin inhibitor? I don't know. But I think something should be done. But let's get back to COVID and what's going on there. Mainly, let's talk about the focus, the mechanisms of action. And I think what we're doing is we're coming out with a investor deck here soon. Am I right, David?
Yes.
Yeah. So when we come up with the investor deck, you can see some of this. So everything that we're talking about is it's out there. You just need to know where to look.
Mm-hmm. Well, that's the key. So, Mike, so where do people need to go look? I mean, at the end of the day, and I don't mean to interrupt you, but
no problem.
You do need investors. I know that's not your primary focus, and I appreciate that also. I think everybody here has been extremely humble. But I just sort of want to be a little direct if I can.
Sure.
Where do people go? So we can go we can go to the stock you're on NASDAQ or Dow?
We're on the over the counter, OTC.
OTC, BIXT. Okay, so they're OTC, BIXT is the symbol. Where else can they go if they are somebody that can help infuse? And I also just want to let you know, I got a text from somebody from Germany that says the vice president should call us. It sounds great what we've been talking about here today. I just want to throw that out there. And they suggested priming the cytosorb with the galectin inhibitors. Just as an idea. So I just wanted to tell you that somebody sent that in. But we need to be able to get people information in order for you guys to continue to do the research that you're doing. But it takes money. We know that. Everything does.
They call Mike, and Mike will give him a document.
They call Mike? Is that how we do it? To get a prospectus, they need to call you?
Yeah.
We do have an effective S1, I believe, and investors should, large investors should know that. If I put on my analyst hat, there's nothing wrong with purchasing the stock in the open market. Again, you have to you consult your financial advisor. I'm not making a buy recommendation here on anything. Again, what I'm doing, my focus is, hey, look, we all know the economics. More buyers and sellers, stock goes up. More sellers and buyers, stocks go down.
Right.
Okay. So.
Right, but these are weird times, though, Mike. Right now we're in the middle of a catastrophe, and the stock market is being influenced by these other things. But go ahead. I'm sorry.
Any investor right now will go and buy stock on the market, help the company eventually to raise substantial amount of money.
So really, just investing in buying BIXT as a stock.
That's what I'm thinking.
Is there any
if there is a large investor, I mean,
well, that's what I'm talking about here. I'm talking about people.
He can come directly to the company. He can call me. Call me.
Okay. So, I don't want to publish your number. So, is there a particular email, web address? Is there some?
They send me an email. I'm the CEO of the company.
So, here's what we need to do. So, take this information down, and then, David, if you could replace BIXT with this email that Dr. Platt's going to give you.
It's david.platt@bioxytroninc.com.
Okay, so let's just spell it out. DAVID dot PLATT at
bioxytroninc.com.
BIOXYTRANINC.com.
Yeah.
Okay, here you go. Let's put that up on the screen if we could, please. Thank you. Go ahead, Mike. I'm so sorry, but I just want to get the information out there because I know the people that watch this program, and I'll get inundated with messages saying, "How do I get a hold of? How do I get a hold of?" And I don't know. I'm not going to give out your phone number. I'm not going to give out your phone number unless that's what you want me to do.
No, they send me an email.
Send an email.
They can mention the program.
Yes, please. In fact, that would be very helpful.
So I will know that the person comes from that program.
I expect all of you. You all better be sending an email immediately. Magic, how many email addresses do we have we can send from? Go ahead. I'm sorry. Mike?
Yeah.
The floor is back to you.
So the other thing that's interesting is remember what let's talk about what David said. The MDX viewer is a tool to get approval, correct?
Right. Now that's also within BIXT.
Yes.
Is the MDX viewer. So your company, just so I understand, your company is diversified platform of the MDX viewer, the BX25, BXT25, and the galectin inhibitor.
Yes.
Okay. Galectin-1 inhibitor specifically. Galectin-1 inhibitor. Okay. Now,
we have two. We have
two.
1 and galectin-3.
Galectin-1 and galectin-3 inhibitor. Okay, very good. I'm sorry, Mike. Go ahead.
So what I wanted to let's just kind of talk about the structure of the company. And the MDX Life Sciences is a private company. So what can for example, your community do? If your community thinks that this is of value and thinks that this is the new frontier, this is something that they want to have. If you're a perfusionist, you got to ask yourself, do I want to have something like the MDX viewer, or maybe something that's a skin sensor? Do I support this technology? If you do, then we probably or David is you want to talk to David about potentially investing in MDX Life Sciences, because that is that's basically a private company, and that private company, MDX Life Sciences, licensed use for clinical trial use of the MDX viewer to Bioxytran Inc., the public company. So that's something that I think perfusionists would have an interest in.
Well, I have an interest, Mike. I mean, for my perfusion colleagues out there, and there's a lot of people that watch this program that aren't necessarily perfusionists. Some of them are physicians, some of them are nurses, some of them are mid-levels, nurse practitioners, and so forth. Some of them are intensive care medicine doctors. So we have a lot of different disciplines that watch our program, not just perfusion, because a lot of what we do translates from the operating room into the ICU to the emergency department for ECLS and for all kinds of reasons. So from a perfusion perspective, having a device like the MDX viewer that I can use in as a bladder catheter, which is already going to go in anyway, so I'm not adding anything to the operation, and I can essentially visualize the effectiveness of my perfusion to that patient, whether or not the microcirculation is staying intact, whether do I need to go up on my flow, down on my flow? Do I need to make an adjustment to the CO2 to shift the oxyhemoglobin dissociation curve? Prediction, right, as a prognosticator of later outcomes is very important. Making sure that we're perfusing the kidneys well enough, that they're going to not suffer. I'm not going to have a patient in renal failure, because that's really a big problem. There's a lot of reasons that we want to know. Measuring the adequacy of perfusion for us is a very rudimentary process of sending out a lab, seeing if we're developing an acidemia. If we're not, then we assume we're fine. It doesn't really give you the whole picture, because so much of that can be affected by what fluids you give, diluting out the lactate, changing the pH just based on the fluid you're administering, bicarb-based, acetate-based, we can go on and on. I mean, there's so many different things we can talk about here. So having that as having the ability to measure mitochondrial health, oxygen supply balance is critical to me being able to do my job. Then when you talk about when we do on-pump, cross-clamp coronary bypass surgery or valve replacements, and we give cardioplegia, which is cold, high potassium, we arrest the heart, we make it cold with the high potassium to depolarize the heart. Now you're telling me I could be infusing a solution that has oxygen in it. I can give it, let a period of time go by. If we're not removing it and it's cold, and we do have potassium in it too, we just add this molecule, we have an arrested cold heart in an oxygen-rich environment. We essentially have no ischemic time. We could stay there for 4 hours. It makes doing a complex operation much safer for that patient from a cardiac stability perspective post-operatively. So, there's a lot of reasons. We have patients who we don't want to give blood to. We're on the heart-lung machine. We know it's got dilutional effect. I'm looking at a low crit. Do I give the patient blood and risk that and give them all of the understood associated risks of blood transfusion, or do I give them the BXT25, get their oxygen carrying and delivery capacity way up, come off bypass, hemoconcentrate the leftover pump in the oxygen in the pump, now the blood in the pump, give that concentrated blood back, and that patient who would have gotten 1 or 2, maybe 3 transfusions of red blood cells gets nothing. You got to think about this from from this. There's so many different pathways. That guys sit here for 2 hours and really not cover everything because there's just so much. Those are the realities of my industry.
So
actually,
one of the things that I wanted to.
And I don't mean to say. I just need to say this one more thing. What about big aortic surgery? What about profound hypothermia, circulatory arrest? When we have to stop we do isolated cerebral
You took
perfusion.
the word out of my mouth.
Oh, I did? But what about those people that we could give a big dose of this to? We could even give it, in the selective cerebral circulation, because what are we there for? I mean, yeah, we want to preserve the organs. We want to preserve the kidneys. We don't want them to go to renal failure. We don't want them to go into, shock liver. We don't want them to have gut ischemia. Yes, all that's true. But the most sensitive organ and the one that will never recover is the brain. That's what we're there for, protect the brain. And when you're doing artificial circulation just by itself, without complicating it with all of these very exotic cannulations for selective cerebral perfusion, we still have a stroke rate of 2 to 3% for routine coronary bypass surgery, 7 to 10% for valve replacement surgery. Now, those may be embolic, but this would help them too. This would help them too. So there's a lot going on here. There's a lot to unpack.
Joe, you should ask David about his experience with his wife and his surgery. It was David. You should elaborate on that.
Yeah, my wife. I told you my wife had a back surgery.
Yes.
And the anesthesiologist said, where I can get this machine? I said, why? Because he said, all I'm doing here is keeping the brain alive.
Yes. Yes.
You give me the tool, I don't need to perfuse any blood. I just keep the brain alive, and I'm happy.
Yes. Yeah. Yeah. I mean, yeah, that's true. I mean, it's kind of true, but you want people to leave the hospital whole. You want their kidneys to work. You want everything to work. I mean, you do. You don't want to just sacrifice the kidneys. But it sounds to me like this has, the kidney is so sensitive. It has such a low PO2. By the time you get down to the glomerulus, the oxygen availability to it is really small. And deficits have tremendous impact. Of course, we all focus on the brain because who wants a stroke? I mean, a stroke is a fate worse than death to most people, Yeah. So, we got to take these things into consideration. And I think anything that we can do to reduce and then of course ARDS, I mean, ARDS is a huge killer in this country. I mean, we all, look, this coronavirus thing is concerning. The graphs that you showed were very concerning compared to H1N1, certainly. But I lived through the H1N1 thing. We had so many people on ECMO, but so many people failed ventilation because their lungs were so stiff and so diffusely infiltrated. They had zero lung capacity. But a system or a tool, a drug like you're talking about, this nanoparticle, that can actually eliminate the need to diffuse the oxygen from the alveolus through the alveolar capillary membrane back into the capillary and be able to move back and forth through that membrane, regardless, is going to mean you can go to lower ventilatory settings. You can rest the lungs more. You can allow them to heal because barotrauma is what hurts them. We don't have that capability because all we have is external mechanical oxygenation, which frequently is not enough, especially for big, super hyperdynamic patients. So this is needed for just simply oxygen carrying capability. Go ahead, Mike. I'm sorry. I didn't mean to take the show over. I do it all the time.
Well, let's ask you a question. How many COVID patients are you treating now with ECMO?
One. In my practice, one.
Okay.
In my practice, one.
So do you see
now
if
COVID
this
negative
gets
after 6 days.
If this gets bad, I hope it doesn't. I think there's some really promising technologies out there, including ours. And I say ours, I mean bioxytrans. I would put that at the top of the list of promising technologies. But you know, there's other drugs out there that that I'm seeing that are going to come to market soon. You know, I'm an analyst, so I look at these things that I think are going to have profound impact on the disease and treat it. Now, the stock market, the one thing that they're not, that everyone was panicking over is human ingenuity. I'm telling you, we as a society, we have it right here. We have the ingenuity, we have the capability to do this. I'm really excited about the technology. I've looked at all the technologies out there. You know, some of the top ones, you know, like remdesivir. You know, that's an antiretroviral.
That's what we gave this guy.
What was that?
That's what we gave our guy.
Okay, it's an antiretroviral, and you know, there's some issues with it, but antiretrovirals, you know, they're designed to work. They're designed to reduce the viral load. But I challenge you. There's an issue with this. What we're doing is we're looking at things the old-fashioned way. Okay, so it's like HIV. Okay, so we're going to let the virus into the cell, and we're going to mess up the process of replicating. That's what an antiretroviral is. And hopefully by doing that, we reduce the spread of the disease. Okay, that's one way to work about it. But what about an entry inhibitor? You know, why are we not using that? For example, you know, there's a great entry inhibitor out there, leronlimab. Okay, that basically stops the HIV virus. It blocks the CCR5. It's one of the co-receptors in HIV. You know, so entry. So and it doesn't have any side effects to it, at least that we know of. You know, there's another thing, and I believe that drug was also being used in a coronavirus in some compassionate use trials. I'm definitely looking to hear more about that. But again, what we're talking about is an entry inhibitor. An entry inhibitor just makes so much more common sense than actually letting the virus in, letting the cell get diseased, and stopping it. You see. And that's why I'm definitely a proponent of the galectin therapy, because with a galectin, what we're going to be doing is we're going to be attaching, the galectins are going to attach to the spikes of the coronavirus. And again, this is theory, guys. We don't have any. We just have scientific journal articles to go off of, but we also have brains and we have common sense. So the theory behind this is it's going to attach to the coronavirus spikes, and there's a specific binding region. It's called the galectin fold, and the inhibitor is going to come in right there and attach. And along with it is this big, huge, clunky galactose side chain, and maybe David could get into that. And it's basically going to cover cover the virus. And now that this virus is covered, what's the liver going to do with it? It wants to process it out of the body and the system. And that's I think one of the key mechanisms of action. All right.
Very interesting. Do you want to elaborate on that?
I published in 2009 how I block. I did crystal. It's not me. My collaborator in the University of Minnesota, named Kevin Mayo. We published couple of articles. It works. It took us about 10 years. 10 years to study the galectin-1 which is on the corona.
On the coronavirus itself. Yeah. You were looking at corona.
No, we didn't look on virus at all.
No.
We just look on galectin-1. And how we can block that lock. How we can adapt the key to the lock. The lock is the galectin-1, and the key was the sugar. So I play with many sugars. You study what is the best sugar, how to do it. Because you see, the problem in this business of carbohydrate, that I can elaborate on that a little bit, and I will explain the background. But in a nutshell, if you don't know what you are doing, your product will be inactive. That's part of the problem, that it's not popular in terms of chemistry. You can find many chemists which are not chemists. You find many biologists, which is very straightforward. You make an antibody, it works. It's the known method. You can make it a lot of it. You make a medication. You can do DNA. You know the sequence. You know how to get it in. You know how to manipulate that. It's quite today. You have CRISPR. You can edit the gene, and people learn how to do it over the years. But I'm not sure you are aware. I want to say to the audience that carbohydrate considered neglected science, and the magnitude of complexity is unimaginable. Carbohydrate as a material to affect biological system only were discovered in the beginning of the 80s, while Nobel Prize on DNA was achieved by Watson and Crick in '56. The lipids were discovered in the '70s with the Nobel Prize around that, and the protein early in the 20th century. Carbohydrate considered neglected science. Even today, there are not many people that are doing carbohydrate chemistry. And the complexity of carbohydrate is enormous.
That's probably why it's neglected.
Not so. I think neglected because, not just because of complexity, because only in the 80s people start to understand why that carbohydrate has a role of fine tuning of recognition. And the complexity of the chemistry is super complex. I cannot describe. I give you an example, which the audience will understand it much better. If, for example, this book, one of the books by American Chemical Society, the How to Design a Drug from Carbohydrate, there are not many drugs are made of carbohydrate. I was the first one to make chemotherapy, non-toxic chemotherapy, anti-cancer, anti-metastasis based on carbohydrate. My previous company is still developing drug for NASH, which is in liver disease, based on carbohydrate. I was the first one to do a company or develop a drug to deliver chemotherapy with carbohydrate specifically. So if you go back to the complexity, I'll give you an example. If you take four amino acids, you know, the building block of protein, and you calculate how many theoretical connections on the computer, and by the way, in real life, in the lab, how many substance or compound I can make with 4 amino acids. Just give it 4 minutes. The number goes to 254. That's it. That's the maximum number. If I, as a chemist, if I know and I make all the crazy connections that I can think about, 254 combinations. 4 sugars give you 32,000 combinations. When we're talking about a carbohydrate to block the galectin-1 for the coronavirus, that sugar has about more than 1,000 sugars in it. There is no computer today. There is no knowledge, there is no science to do it. I was struggling myself to understand how to manufacture that. It's not simple because when you manufacture by the standard of the FDA, you need to know exactly what is the structure. You need to repeat it. Very difficult. And that's what I actually enjoying the complexity, because probably I'm a little bit not stable mentally.
I didn't say it. I didn't say it. I'm not the one who said that.
To go only to the dark side of the moon, so to speak.
I don't have that kind of patience. Thank God we have people like you. That's all I can say.
No, it's a complex. It's insane in many ways. You deal with complexity of and diversity that it creates a lot of manufacturing problem and experiment that doesn't work, and it's all related to the chemistry. I was struggling for many years, for example, connecting to the heme. I was not, I didn't understand why suddenly I'm doing the same thing, it doesn't work. So I figure out why it didn't, but it takes time to understand. It's not simple, that's what I'm saying.
Well, 30 plus years. Mike, would you like to finish out with any thoughts? I'm gonna be sending this out to some folks. You're probably gonna get some emails. Would you like to add your contact to this, or are you happy with it all going to Dr. Platt?
No, it's fine. It can all go to David. I'm very excited with the technology, and I really hope that everyone that listens to this presentation, really, I want to get a sense of awareness out there that we can really do something with galectins. Galectins are very important. I think they're absolutely crucial to the coronavirus. And I mean that above all the other technologies that are out there. I'm really, as an analyst, I'm putting on my hat as an analyst, I'm saying this is one of the most exciting technologies out there. Now remember, with inherent with drug development is a lot of risk, okay? So we could fall flat on our face, but when you look at all the things that we can do, you got to ask yourself, does it really make sense that this company is going to fall flat on its face when we have so many different indications that we can go after? From that perspective, I want to say it's hard to we really got to screw up bad to fail at this. So that's what makes me really excited. But on the galectins front, people need to know about this. People need to know that, well, geez, if I am positive, it's pretty intelligent to know what my galectin levels are. These are common sense things, and I hope someone that's listening to this gets this to Dr. Fauci. This is crucial. This is crucial, and that's really the message that I want to get through, is that we need to be talking to these guys. This is very important technology.
Yeah, especially since they're talking
awareness
about...
is the most important thing. Yes, we need money. Yes, you can buy the stock. Yes, you can contact David. There's an active offering out there. So yes to all of that, but let's yes to awareness.
Yes,
we need to be aware of this.
And I appreciate that so very much. And I think you're 100% right. In the current environment of looking to private industry, which is the current philosophy to solve these problems, because government can't solve them, private industry can. I think you're 100% right. They need to get this information to the people who can direct some of these compassionate use trials to get this up and going. From my perspective, though, and I appreciate your passion for the current situation with Corona and COVID-19, I will tell you that I am profoundly worried about the rest of the world, the rest of the people, the rest of the population, the rest of our citizens, both here in the United States and elsewhere, who are still going to have their typical health-related problems. So from my perspective, the MDX viewer, the BXT25, and then also the galectin inhibitor for the corona combined is something that I take really most seriously, that you're not one-dimensional. This is a very strategically diverse organization, company that is looking at all facets and other than one group of patients, which eventually will go away. This problem, it may rear its head again sometime, but the heart disease, we've had 647,000 deaths in this country last year from heart disease. 647,000.
There are about 900,000 stroke.
900,000 strokes.
A year.
Right. A year. And the
That's
cost
that's our market.
and the cost to society
Huge.
is enormous. So, you know, people that are sick, that have perfusion deficits in the ICU, that have oxygen deficits to their tissue, that go into renal failure, end-stage renal disease, and then need to be on dialysis. Enormous.
It's a game changer.
It's a game changer. It's the galectin. I know we're focused on that because that is the hot topic, but I can't ignore that we have many, 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 many sick people that can't be just left to their devices in preference to patients who have coronavirus. That's my feeling.
By the way, you know where else galectin 3 and galectin 1 play a major role in recognition? Influenza.
There you go.
Yeah, there's a universal target on the COVID spikes, and in that article that I mentioned, it's actually mentioned in our press release. What happens is it's really interesting, and I don't want to take too much time, and I don't want to belabor the point, but the virus evolved from galectins, and that's basically what the journal articles talked about. What came first? The first recognition domain was the galectin, and then what came on top of it, some people might have heard about the ACE2 receptor. Well, that's right on top of the galectin on the recognition domain.
Yeah.
And what happens is the galectin recognition domain is right over here. It's on the side. So a normal antibody can't, it's Y-shaped, right? So it can go here on top of the ACE, but it can't go here. Okay, it's just too big, and that's what the research has indicated.
That's why I call it fine tuning of recognition. The antibodies,
right.
It's not. Antibodies are too big. The carbohydrate is very small in terms of distance. Look, the whole of the virus is like what, 20 or?
No, it's teeny. Yeah,
30 nanometers.
Beyond microscopic.
Yeah, it's like very small.
Yes.
So you need only sugar can be small enough because it's smaller than the virus. The recognition, the piece that recognize in terms of distance, so only sugar. But antibodies don't have that.
Very interesting. Well,
those are fact. Those are fact. This is not me. This is a fact, scientific fact.
Absolutely. Well, I need all of the press releases, please, so we can put those all in one place, attach everything to this. The journal article, I need that to put it also in there. We'll get it, though, if you send us the releases. We'll get all of that. We're going to package all this together, put it back up on the web in edited form, make sure that it is, we fix the glitches that we had and all that kind of thing to make it so that it's more seamless. So we'll take care of all of that stuff. We just need the information. If you can send it to me via email, it'd be appreciated. I would like to, if I could, close out and thank, of course, Dr. Majewski. Hopefully he's sleeping, I hope. I want to thank you, of course, Dr. Platt, for your generous time in coming here all the way from the East Coast and taking a flight to do this. Very important information, and I can't tell you how much I truly, deeply appreciate it on behalf of myself and our audience. Mike, thank you for, coming in via Skype, for giving us your insight and your thoughts. Everything is appreciated. At the end of the day, this is about information sharing, about getting information out there, and that's how we all have an impact in the progress of helping the human experience fighting diseases of all sorts. And that's information sharing and really smart people like yourselves and Dr. Majewski. I mean, really brilliant people, and I appreciate you a lot.
Thank you. Thank you.
You're welcome.
Thank you.
Thank you, Mike. Okay. Saturday, we have a program on Saturday. So for those of you who are on, who have signed up for this PerfWeb 34, this is day 2. We'll go to day 3 on Saturday with Stephanie Ibus's program on women in perfusion, which is really about women in high demanding jobs. It's going to morph, as you well know. But in healthcare, you know that we have a special responsibility, being on call, called out all hours. It's very challenging in order to be able to start a family and manage your personal life with your professional life. Anytime you're in these kind of high-level demanding jobs, we're going to have an incredible faculty. Ann Greco, I know, is coming with the American Board. Several other people. I can't remember everybody's name right now. I'm just blanked out. But it's going to be a great program. We'll see you on Saturday morning, I believe, at 7:45 or 8 o'clock. Thank you all again for your time and attention tonight. We appreciate you. Without all of you, we wouldn't have a show or a program or a need to be here. So I'm indebted to all of you. And again, Dr. Platt, I can't thank you enough. This was very, very, very generous of you and your time and your expertise and knowledge. You, in my view, you're a genius. Okay, you're a genius. I don't
see it this way.
I know tubing. I know tubing and clamps and flow and all that kind of stuff. When you start getting into all of this really deep science and carbohydrates, I'm just amazed at how much a person can pack inside their head. Because I can tell you right now, this is mind-boggling to me. Mind-boggling. It's been very informative, but very, very, very serious. It's a serious subject.
Yeah.
Mm-hmm. So thank you again. See you all on Saturday.
The point of care program at Huntsville Hospital is approaching its 20-year anniversary. As we began to grow as a hospital and provide more complex services to our community, the need for point of care testing became very clear in our organization. Many of our departments consider the EPOC device to be crucial to their ability to quickly move their patients from entry to the hospital to procedural room. It helps them monitor the patient's status during their procedures and post-procedure, so it's very integrated into our entire continuum of patient care. If you have any patient population where blood management is crucial, then a point of care device is very useful in those types of settings. It can provide a quick result on a very small amount of blood, and that is really important in some of our patient population.
We are starting a new process where we're going to screen our patients on the floor for sepsis. The patient can look and actually feel okay. They will show us signs and symptoms of sepsis. So what we do when they show these signs, we get a lactate level.
Lactic acid is a product of the sepsis cascade. So with sepsis, the inflammatory light switch goes on, the cascade starts churning, and unfortunately, once it starts going, we have to stop it. It will not stop on its own. The faster the lab draw, the faster the lab results, the faster you can act on it.
The process in the lab is what the process is. It just takes a certain amount of time for the instruments to run those tests.
I mean, we're talking 30 minutes even on a stat because that's the process. That's just how long it takes. There's not really anything you can do about that. For every hour you wait to give an antibiotic, the mortality for that patient increases 8%. So now that we have the handheld device, the nurse can get those results within a few minutes, and we know how to treat that patient. If a patient is going into septic shock, we do not have minutes to spare. We have got to get it done quickly. So when you get the little beep from the EPOC that says your results are in, you go with it. You don't even think twice about it. You're like, this is good. This is what I'm going to do for this patient. The next steps are clear.
I think our mortality on the initial floors dropped by almost 50% in the first 3 to 6 months. At this point, we're about hospital-wide. I think we'll be hospital-wide another 2 to 4 months. So this has become a huge program within our system.
Without the EPOC device, it would really be hard for us to get everything done in the timely manner. So it's really helped us to get our program going.
I mean, whether you're a surgeon, whether you're a medicine doctor, whether you're a dermatologist, you want to be able to do things in a timely fashion so that you can be most effective.
This really makes a difference in patient outcomes.
Mixed cannulae, expanding performance standards. High-performance cannulae for direct and femoral cannulation in minimally invasive cardiac surgery. LivaNova's complete range of mixed dedicated products allow easy and reproducible procedures. The portfolio includes the sutureless aortic valve Percival, Memo 4D for mitral valve repair, the HeartLink system for assisting the perfusionist in applying goal-directed perfusion therapy, and a broad choice of high-performance dedicated cannulae. LivaNova's range of cannulae suit every cannulation technique for minimally invasive cardiac surgery. Venous and arterial femoral cannulation can be performed using the WRAP venous cannula and the EasyFlow Duo. For arterial direct and venous femoral cannulation, the EasyFlow aortic cannula and the WRAP are available. For venous femoral access, the LivaNova WRAP cannula features a dual-stage configuration for optimal drainage of the inferior and superior vena cava without interfering with the right atrium. The high-flow dynamics of WRAP improves the capability of drainage with a low pressure drop. An optional Sellinger dilator kit is available to perform percutaneous insertion. The LivaNova EasyFlow Duo cannula is designed for a safe and smooth femoral arterial cannulation. Its conical dispersion tip design allows a more gentle flow in the femoral artery, and its red cap avoids blood backflow during obturator removal. A dilator kit for percutaneous insertion is also included with the cannula. EasyFlow cannulae are designed for safe, reproducible, direct aortic cannulation and include a malleable stylet to aid cannulation insertion, facilitating bloodless introduction into the aortic arch, as well as a special conical dispersion tip for a more gentle flow in the vessel to reduce shear stress.
Like all perfusion professionals today, we are busy and frequently short-staffed. Does taking time off for a conference create a burden for your teammates? Has reimbursement for your continuing education dropped? Have you priced the cost of going to a meeting lately? Registration fees, airfare, transportation and parking, hotel, and meals out? This totals up to having an out-of-pocket expense of over $2,500. And then you hope the content is good. Perfusion Education has heard your concerns. They have a simple and seamless system that works on any platform, computer, tablet, or even your phone. You can chat during the program or even phone in and be live on the air. The content, second to none. Perfusion Education is by perfusionists and for perfusionists. Create a free account and check us out today.